mulai?
Dan kaumnya nggak usah divert one to one.
Oh,
Nggak usah divert.
apa?
Coba one to one, coba immediately. Yang 13 aja tapi. Yang 13 kan yang kemarin itu mingguan udah buat dan kan ada 2. 1, 4. Nah, itu udah confirm. Yang biasa aja kan. Yang 13 itu maksudnya trip itu kayak on freeze.
Siap.
Cek 1, 2, 3. Selamat pagi Bapak Ibu yang terhormat. Kami persilakan untuk memasuki ruangan acara. Sebelum itu kami persilakan juga Bapak Ibu untuk menikmati hidangan coffee break yang
Maksudnya?
telah kami sediakan di samping ruangan. Acara akan dimulai pada pukul 9 waktu Indonesia Barat. Jadi kami persilakan Bapak Ibu untuk dapat menikmati terlebih dahulu coffee break sajian yang telah kami sediakan di samping ruangan. Kami mengajak Bapak Ibu peserta hadirin pada hari ini untuk mengisi kursi yang berada di depan terlebih dahulu. Sekali lagi, kami mengajak Bapak Ibu hadirin pada hari ini untuk mengisi kursi yang berada di meja depan terlebih dahulu. Tes 1 2 3 cek.
Tes, tes, tes 1 2 3. Tes, tes 1 2 3. Tes, tes 1 2 3. Tes,
Cek, cek, cek. Berarti harus
tes, tes
cek
1 2 3. Cek,
1 2 3.
cek 1 2 3.
Fadli udah jalan ya? Kakak Dika jam 2 tuh aku dijanjian sama Mbak Rina.
Cek.
Pemeriksaan.
Bapak Ibu peserta workshop yang kami hormati, acara sebentar lagi akan kita mulai. Dimohon Bapak Ibu peserta workshop untuk dapat memasuki ruangan acara.
Itu pada itu ya Aspi ya? Aku boleh.
Bismillahirrahmanirrahim. Yang terhormat Direktur Registrasi Obat Badan Pengawas Obat dan Makanan, Dr. Ya Christine Siagian, SSI Apoteker MSC.
Yang kami hormati narasumber pada hari ini, Dr. Rernat Apoteker Aloysia Anita Artarini SSI MSc dari Institut Teknologi Bandung, Profesor Dr. Amin Subandrio PhD, Spesialis Mikrobiologi Klinik Konsultan, Ketua Komite Nasional Sel Punca Indonesia.
Yang kami hormati Ketua Tim Kerja di Lingkungan Registrasi Obat Badan POM,
perwakilan industri farmasi dan universitas yang hadir secara luring maupun daring,
seluruh peserta workshop yang berbahagia, baik yang hadir di tempat ini maupun yang bergabung secara daring. Selamat pagi Bapak Ibu, mohon izin untuk membuka acara. Kami akan menyampaikan sebuah pantun, jadi mohon untuk diikuti dengan kata cakep ya Bapak Ibu. Belum.
mentari terbit belumlah tinggi,
Cakep.
membawa sekop pergi bekerja.
Cakep. Senang sekali kita berjumpa lagi di Workshop Elevate hari kedua. Boleh kita beri tepuk tangan? Assalamualaikum warahmatullahi wabarakatuh.
Selamat pagi dan salam sejahtera untuk kita semua. Puji dan syukur kita panjatkan kehadirat Tuhan Yang Maha Esa karena atas rahmat-Nya, kita dapat hadir dalam keadaan sehat walafiat di tempat ini pada hari Rabu, 8 Oktober 2024 dalam acara
Elevate, Empowering Regulatory Excellence, Workshop on Advanced Therapy Medicinal Products. Boleh tepuk tangannya Bapak Ibu? Bapak Ibu yang berbahagia, sebagaimana yang telah disampaikan oleh Kepala Badan Pengawas Obat dan Makanan di hari pertama, bahwa workshop Elevate ini merupakan sebuah bentuk komitmen Badan POM untuk mendukung pengembangan advanced therapy medicinal product di Indonesia.
Di hari pertama, kita telah sama-sama mendapatkan ilmu dan penjelasan terkait kerangka berpikir, tantangan, dan kesempatan dalam ATMP dari perspektif Badan POM sebagai regulator yang mengawasi obat di Indonesia. Selain itu, juga telah kita mempelajari terkait fungsi GMP dalam pengembangan ATMP.
Pada hari ini, kita akan sama-sama mendapatkan kesempatan mendengarkan dan berdiskusi langsung dengan pakar akademisi dan regulatori dari negara lain terkait advanced therapy medicinal product ini, yang pasti akan sangat sayang untuk dilewatkan.
Untuk itu, tidak berlama lagi, selanjutnya masuk kepada sesi workshop pertama di hari kedua dengan materi pertama yaitu Advanced Therapy Medicinal Products and Overview.
Dan dilanjutkan dengan materi The Role of Academia in the Development of ATMPs. Sesi workshop akan dipandu oleh moderator, Ketua Tim Kerja Penilaian Uji Klinik dan Obat Pengembangan Baru, Ibu Ati Ratnawiyati, SSI Apoteker MFP. Untuk itu, kepada Ibu Ati, waktu dan tempat kami persilakan.
Terima kasih MC. Jadi Bu, izin duluan. Nanti menyusul. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi Ibu dan Bapak semua. Hari kedua training Elevate ya. Yang sangat menarik dari hari pertama. Ibu dan Bapak semua masih tetap semangat ya? Ya, tetap semangat dan siapkan pertanyaan-pertanyaan terbaik untuk para pakar yang sudah hadir hari ini gitu ya. Nanti kita akan berdiskusi supaya hidup forumnya. Baik Ibu dan Bapak sekalian, saya Ati Ratnawiyati akan menjadi moderator untuk sesi pagi di hari kedua training ini. Selama kurang lebih 1,5 jam ke depan, kita akan mendapatkan pemaparan dan berdiskusi tentang overview of ATMP dan the role of academia in the development of ATMPs. Untuk itu, mari kita sama-sama sambut kepada yang terhormat Prof. Amin dan juga Ibu Dr. Anita untuk maju ke panggung. Silahkan Prof. Ya, silahkan applause-nya. Ya, baik, terima kasih Prof. Amin dan juga Bu Anita. Jadi sesuai dengan topik kita ATMP ya, sudah dibahas kemarin dalam sesi pertama training ini, bahwa perkembangan ATMP menghadirkan banyak peluang baru untuk pengobatan berbagai penyakit kronis, genetik, bahkan untuk penyakit yang belum ada obatnya. Namun juga ada berbagai tantangan dalam pengembangannya, baik dari sisi ilmiah, klinis, maupun regulasi. Salah satu aspek yang sangat penting adalah kolaborasi ya, antara sektor akademik, kemudian industri, dan juga regulator untuk mempercepat inovasi di bidang ATMP ini. Hari ini kita akan mendapatkan pemahaman yang komprehensif tentang ATMP serta menggali lebih dalam bagaimana peran akademia dalam pengembangan ATMP. Bersama kita sudah hadir Prof. Amin dan juga Ibu Anita, yang Bapak Ibu semua mungkin sudah kenal betul beliau-beliau ya, tapi izin kalau jadi moderator tetap harus membacakan CV-nya ya. Ya baik, Bapak Ibu, sebelum kita mulai diskusi, izinkan saya untuk membacakan kurikulum vitae singkat para narasumber yang sangat berkompeten di bidangnya. Yang pertama akan menjadi narasumber adalah Ibu Dr. Rernat APT Alusia Anita Artarini SSI MSc. Beliau menempuh pendidikan farmasi dan masternya di ITB, kemudian master di bidang molekular medicine ya Bu ya, di Charité University at Medicine Berlin. Mudah-mudahan nggak salah bacanya ini. Ya, di Jerman, kemudian lanjut masih di Jerman ya Bu ya, PhD-nya dari Humboldt University of Berlin. Saat ini beliau sebagai asisten profesor di Sekolah Farmasi ITB. Kemudian narasumber kedua adalah Profesor Dr. Amin Subandrio, PhD, SPMKK. Beliau mendapatkan dokter dan spesialis mikrobiologi klinis di Universitas Indonesia. Kemudian PhD-nya dari Osaka Kobe University, Jepang, serta menjadi profesor di Universitas Indonesia. Saat ini Prof. Amin menjabat beberapa posisi penting sekaligus nih. Banyak ya. Saya sebutkan beberapa di antaranya, yaitu sebagai Ketua Komite Pengembangan Sel Puncak, the Chairman of Steering Committee Asian Partnership for Emerging Infectious Disease Research, serta Head Research and Community Service Institution di Universitas Pancasila. Ya, demikian CV beliau-beliau, para narasumber. Tanpa memperlama ya, supaya sesi pemaparan dan diskusinya lebih intensif, kita tidak berlama-lama lagi, Ibu. Silakan, kami persilahkan Ibu Anita untuk memberikan pemaparan. Waktu kita 30. Ya.
Baik, terima kasih Ibu atas introduction-nya gitu ya. Selamat pagi Ibu Bapak semua, baik yang hadir offline maupun online. Mungkin pada kesempatan hari ini saya hanya akan memberikan gambaran ringkas gitu terkait dengan advanced therapy medicinal product ini apa sebetulnya. Dan isinya mungkin akan lebih banyak contoh-contoh dari produk-produk yang sudah approved gitu ya di dunia gitu. Mungkin bisa next slide. Ya, kalau kita lihat dari cakupannya, ini saya mengambil definisi dari European Medicines Agency, tapi kalau yang seperti kita tahu di CPOB 2024 juga sudah masuk di Annex 2A gitu ya, terkait dengan advanced therapy dan mengacu ke PICS gitu. Nah, itu tidak terlalu berbeda jauh gitu ya. Dia mencakup di situ ada gene therapy atau terapi gen, kemudian ada cell therapy, dan ada tissue engineered medicines. Nah, mungkin nanti saya akan cerita kalau terapi gen ini macamnya bisa apa saja gitu ya. Kalau sel terapi contohnya seperti apa, dan kalau tissue engineered medicines ini contohnya seperti apa gitu. Jadi mungkin lebih supaya lebih ada gambaran, dan kalau kita mau mengembangkan itu alternatifnya masih banyak sekali ya. Next slide. Nah, ini contoh yang direkap oleh European Medicines Agency dalam 10 tahun terakhir. Memang yang paling banyak dikembangkan adalah gene therapy medicinal product. Ada beberapa sel terapi di situ seperti Epvalo gitu ya, yang cenderung lebih baru, dan ada ada 2 produk tissue engineer therapy. Tapi ini hanya yang disetujui di European Union. Jadi rekapnya yang disetujui di US FDA itu belum masuk ke dalam rekap ini. Dan seperti kita tahu di akhir 2023 itu ada gene therapy yang baru di-approve, dan dia menggunakan teknologi CRISPR-Cas yang relatif lebih baru gitu ya. Topiknya menjadi salah satu pemenang Nobel Prize di tahun 2023. Next slide. Untuk pertama kita masuk ke topik gene therapy, ya intinya dia adalah membawa suatu gen gitu ya, jadi menyisipkan suatu rekombinan DNA ke tubuh gitu untuk memproduksi atau untuk memperbaiki kondisi suatu penyakit. Bisa untuk penyakit genetik, tentu saja kalau kita kenal terapi gen banyaknya untuk penyakit genetik, tetapi tidak menutup kemungkinan untuk penyakit lain seperti kanker gitu ya, dan sebagainya. Nah, next slide. Kalau kita lihat modifikasinya bisa berupa penambahan gen gitu ya, memperbaiki dari suatu misalnya gen yang termutasi diperbaiki menjadi yang benar, atau dia lebih ke arah inhibisi contohnya. Jadi ada ekspresi protein berlebihan diinhibisi gitu ya, atau diedit. Nah, ini misalnya dengan sistem CRISPR-Cas itu memungkinkan adanya genome editing. Kemudian bagaimana gen ini didelivery atau dihantarkan, bisa menggunakan viral vector atau menggunakan non-viral vector. Lalu dapat diberikan secara in vivo maupun secara ex vivo. Nah, contoh dari yang pemberian secara ex vivo ini mungkin Ibu Bapak lebih sering mendengar terkait dengan CAR T-cell therapy atau chimeric antigen receptor T-cell therapy. Next slide. Dari berbagai produk yang sudah pernah disetujui sebelumnya, kalau kita lihat dari cara deliverynya, sampai sejauh ini paling banyak menggunakan sistem viral vector. Tetapi ini tidak menutup kemungkinan pengembangan dengan cara non-viral vector gitu ya. Karena yang Casgevy itu dia nanti kalau saya cerita di belakang, dia tidak menggunakan viral vector untuk delivery-nya. Dari viral vector yang dipakai itu dominannya menggunakan AAV atau Adeno Associated Virus atau menggunakan lentivirus. Nanti saya akan cerita atau sharing sedikit gitu ya, apa sih AAV dan lentivirus ini. Kemudian dari segi pemberiannya ke pasien itu bisa dengan cara in vivo ataupun dengan cara ex vivo. Next slide. Kalau kita lihat apa bedanya in vivo dengan ex vivo, untuk gambaran sedikit gitu ya Ibu Bapak. In vivo berarti kita langsung deliver produknya ke pasien gitu. Kalau dia gene therapy dibawa oleh adeno associated virus, maka produk ini yang kemudian disuntikan atau diberikan ke pasien untuk kemudian memperbaiki penyakit yang ingin diobati. Sementara kalau ex vivo itu berarti ada sel dari pasien yang dikeluarkan terlebih dahulu, kemudian sel ini dimodifikasi di luar tubuh gitu ya, bisa dengan viral vector atau menggunakan teknologi non-viral vector, kemudian diperbanyak selnya dan sel-sel yang sudah berhasil termodifikasi ini yang akan dikembalikan lagi ke pasiennya. Jadi ini bedanya in vivo dengan ex vivo delivery. Gitu. Tergantung case-nya yang ingin yang ingin dicapai seperti apa gitu ya. Untuk ex vivo sendiri mungkin bisa di next slide. Ya, untuk sel yang dikeluarkan dari donor atau dari pasien ini ada dua istilah yang banyak dipakai, autologous versus allogeneic. Untuk autologous berarti selnya berasal dari pasien itu sendiri, dikeluarkan gitu ya, kemudian ditreatment di luar, lalu nanti dikembalikan lagi. Sementara untuk allogeneic therapy, ini nanti ada contohnya di bagian yang cell therapy, dia dari donor yang unrelated sebetulnya. Jadi donornya bisa siapa saja, orang sehat mungkin gitu ya. Kemudian nanti selnya akan diberikan ke pasien yang membutuhkan. Jadi nanti ketika melihat dari produknya bisa terlihat dia autologous atau dia allogeneic therapy. Next slide. Nah, ini salah satu contoh yang di delivery secara in vivo. Contoh produknya adalah Voretigen Neparvovec, mungkin Ibu Bapak lebih kenal dengan nama Luxturna. Dia adalah adeno-associated virus tipe 2 yang membawa DNA pengkode RPE65. Jadi digunakan untuk mengobati retinal dystrophy yang diakibatkan oleh mutasi pada gen RPE65. Jadi dia jadi vektornya membawa DNA pengkode RPE65 yang benar gitu ya, sehingga nanti di dalam sel kalau produk ini disuntikan ke pasien, di dalam sel dia akan mengekspresikan protein RPE65 yang urutannya benar sehingga mengembalikan fungsi dari sistem penglihatannya gitu ya untuk mengatasi penyakit genetik. Next slide. Kalau kita lihat adeno-associated virus vector itu apa? AAV ini berbeda ya Ibu Bapak dari adenovirus vector. Jadi dia dua virus yang berbeda gitu. Jadi bukan adenovirus. Kalau adenovirus dia virus dengan materi genetik double stranded DNA. Sementara AAV ini adalah virus dengan materi genetik single stranded DNA. Namanya adeno-associated virus karena adeno-associated virus ini kalau kita produksi atau diperbanyak, dia memerlukan beberapa komponen dari adenovirus gitu ya. Secara genom cukup simpel, jauh lebih kecil dari adenovirus gitu ya. Dan kalau kita mau memproduksi AAV rekombinan, kita bisa menggunakan beberapa cara, tetapi salah satu cara yang paling mudah adalah menggunakan 3 plasmid yang terdiri dari transgene, kemudian ada helper plasmid, dan ada packaging. Ya, packaging ini biasanya untuk mengkode protein-protein yang menjadi struktur dari vektor virus itu sendiri, gitu ya. Jadi tadi komponen dari adenovirus yang dibutuhkan untuk replikasi AAV dibawakan di dalam helper plasmidnya. Lalu kalau kita transfeksi 3 plasmid ini ke dalam sel, misalnya sel HEK293, kita bisa memproduksi adenovirus vektor ini. Dan pada kasus seperti Luxturna tadi, maka gen RPE65-nya itu dibawakan di dalam transgene plasmid yang nantinya akan dikemas di dalam struktur atau di dalam AAV-nya. Next slide. Nah, ini tadi salah satu contoh gene terapi sebetulnya. Kalau di MA, CAR T-cell itu masuk ke dalam gene therapy medicinal product. Meskipun walaupun ada melibatkan sel gitu ya di situ. Tetapi jadi sel ini diambil dari pasien karena dia sistemnya autologus. Jadi diambil dari pasien, disortir, diambil T-selnya saja, kemudian T-selnya dimodifikasi menggunakan lentivirus ya supaya dapat mengekspresikan yang namanya chimeric antigen receptor. Kemudian sel T yang berhasil mengekspresikan CAR, chimeric antigen receptor ini diperbanyak dan kemudian dikembalikan lagi ke pasiennya untuk mengobati kanker sebetulnya. Jadi untuk membunuh sel kanker. Kalau kita lihat next slide dari produknya ini yang mungkin Ibu Bapak banyak mendengar itu ada Kymriah dan Yescarta. Ini untuk imunoterapi. Jadi sel T-nya dimodifikasi memiliki chimeric antigen receptor. Jadi chimeric antigen receptornya itu yang ada di sisi sel T-nya gitu ya. Di sisi sel T-nya. Nanti di bagian reseptor yang menjulur keluar itu ada SCFV, single chain variable fragment, yang akan mengenali protein CD19 dari sel limfoma, sel B limfoma yang akan ditarget gitu ya. Kymriah dan Yescarta ini hanya berbeda di urutan dari reseptor yang dimodifikasi sel T-nya. Jadi nanti kalau sel T-nya berhasil mengekspresikan atau dimodifikasi membawa SCFV yang mengenali CD19 ini, dia di dalam ketika dikembalikan ke pasiennya di dalam tubuh pasiennya itu akan mengenali atau mentarget sel B yang memiliki CD19 dan kemudian membunuh sel B limfoma yang CD19 positif. Nah, next slide. Kenapa dipilihnya lentivirus vector? Karena lentivirus vector ini termasuk dalam keluarga retrovirus, di mana dia di dalam sel yang ditarget, dia mampu menyisipkan DNA yang dibawakan di dalam virusnya ke dalam genom sel inangnya. Jadi kalau tadi kita modifikasi sel T-nya dengan chimeric antigen receptor, maka chimeric antigen receptor ini akan tersisipi di genom sel T dan kemudian sel T-nya bisa mengekspresikan chimeric antigen receptor ini terus-menerus sehingga ketika lentivirusnya diberikan atau sel T-nya ditransduksi dengan lentivirus rekombinan ini, dia diperbanyak kemudian diberikan ke pasien, nanti di dalam tubuh pasiennya bisa mengenali sel B yang CD19 positif gitu ya. Jadi ada dua sisi nih, produksi lentivirusnya sendiri dan kemudian bagian modifikasi sel T di mana sel T-nya diambil dari pasien itu sendiri. Oke. Next slide. Nah, ini gambarannya yang terkait dengan masih dalam topik gene therapy. Ini yang di-approve akhir 2023, Casgevy dan Lyfgenia ini dua-duanya sama-sama mentarget sickle cell disease. Hanya yang berbeda, Casgevy menggunakan pendekatan CRISPR-Cas system gitu ya. Jadi sel hematopoietic stem cell dari pasien dikeluarkan, ya lalu kemudian disortir CD34 positif dan ada editing gen BCL11A. Ya, tujuannya untuk meningkatkan produksi fetal hemoglobin yang secara struktur masih benar sehingga mengurangi atau menghasilkan tidak mengobati dari sickle cell disease-nya. Sementara untuk Lyfgenia yang sisi kanan Ibu Bapak diambil dari pasiennya adalah HSC dan dia dimodifikasi seingat saya menggunakan AAV gitu ya. dengan membawakan gen beta globin yang benar gitu. Jadi pendekatannya ada dua, sama-sama mengobati sickle cell disease, kemudian tetapi yang leukemia dia gen yang dibawa adalah beta globin, sementara yang Casgevy dia yang dibetulkan adalah sistemnya dengan gene editing gitu ya. Nah, ini menarik karena gene editing gitu prinsipnya adalah dia akan mengedit bagian dari gen yang kita inginkan. Nah, kok beda Bu, bukan beta globinnya gitu ya, tetapi BCL11A. Next slide. Nah, ternyata di gen BCL11, jadi gen BCL11A ini merupakan protein yang menjadi transcriptional receptor untuk gamma globin. Jadi kalau BCL11A terekspresi dalam jumlah banyak, maka ekspresi dari gamma globin dia menurun. Padahal kita ingin meningkatkan ekspresi dari gamma globin di pasien dengan sickle cell disease tadi. Jadi promotor dari bagian BCL11A ini ditarget menggunakan CRISPR-Cas gitu ya. CRISPR-Cas itu sistemnya ada di gambar yang di bawah, di mana kita menggunakan guide RNA gitu. Guide RNA akan mengenali pump sequence gitu, dan kemudian dia akan merusak, mengedit urutan DNA di posisi guide RNA beberapa nukleotida sebelum pump. Dan di situ ada GATA1 binding site. Jadi GATA1 binding site ini dirusak sehingga transkripsi dari gen BCL11A-nya tidak terjadi. Kalau transkripsi BCL11A tidak terjadi, maka ekspresi BCL11A tidak ada lagi inhibisi ekspresi dari gamma globin gitu ya. Mungkin agak pusing gitu ya mengenali CRISPR-Cas ini, tapi dia prinsipnya adalah gene editing. Tinggal yang mau kita mau kita rusak atau mau kita betulkan itu gimana. Jadi CRISPR-Cas ini macam-macam, kita bisa introduce insertion atau deletion gitu ya, atau kita mau ganti basanya. Kalau mau diganti basanya, maka pada sistem CRISPR-Cas ini bisa dibawakan urutan yang benarnya untuk mengganti. Tentunya spesifisitas dari editing ini harus dipastikan berjalan secara spesifik sehingga tidak menghasilkan off-target effect. Off-target effect itu maksudnya dia mengedit di daerah yang tidak kita inginkan gitu ya. Dan mungkin kalau Ibu Bapak mengikuti, kemarin sore baru diumumkan Nobel Prize in Medicine, itu topiknya terkait dengan microRNA. Nah, microRNA ini juga salah satu non-coding RNA yang bisa meregulasi ekspresi dari suatu protein. Jadi one of teknologi yang bisa kita pakai untuk sebagai bagian dari gene therapy. Next slide. Mungkin itu yang terkait dengan terapi gen. Waktunya masih berapa menit lagi? 5 menit lagi. Saya cepat aja terkait dengan sel terapi gitu ya. Somatic cell therapy. Nah, ini mengandung sel yang telah dimanipulasi untuk mengubah karakter biologinya gitu ya, sehingga dapat menghasilkan fungsi yang kita inginkan. Saya beri satu contoh produk saja di sini adalah Evalo. Next slide. Langsung aja Mas ke yang setelahnya. Nah, Evalo ini menarik sebetulnya karena ini adalah allogeneic T-cell immunotherapy untuk pasien dengan Epstein-Barr virus positif post-transplant lymphoproliferative disease. Jadi pada kasus-kasus tertentu pada pasien yang menerima transplantasi gitu ya untuk kondisi penyakit ini, dia mengalami kondisi yang ada terpengaruh oleh keberadaan EBV atau Epstein-Barr virus. Nah, yang menarik sebetulnya adalah karena ini adalah allogeneic T-cell therapy. Jadi donor selnya bukan dari pasiennya tapi dari orang lain ya. Mungkin kita lihat sedikit bagaimana mekanisme kerjanya. Next slide. Jadi dari donor, makanya dia di tahap pertama disebutnya third party gitu ya, orang lain unrelated gitu. Yang diambil adalah PBMC. Yang diambil ada dua, di bagian yang ke atas itu adalah sel T, kemudian yang di bawah, sorry salah, yang di atas adalah sel B, kemudian yang di bawah itu adalah sel T. Nah, sel B yang diambil, jadi dipisahkan sel B dari sel T-nya, kemudian sel B yang di atas, betul kan ya? Betul. Yang di atas sel B-nya dipilih. Ya, benar. Ya, sel B-nya itu diinduksi menjadi antigen presenting cell, gitu ya. Dan kemudian setelah sel B diinduksi menjadi antigen presenting cell, dia akan ditemukan oleh sel T yang spesifik EBV. Gitu. Dia akan ditemukan dengan sel T yang spesifik EBV dan sel T-nya ini akan teraktivasi dan kemudian diekspansi gitu ya. Ketika teraktivasi, dia perlu diseleksi dengan kecocokan HLA dari pasiennya. Nah, jadi di tahap akhir itu ada karakterisasi sel T. Jadi sel T-nya diaktifasi oleh sel B yang diinduksi menjadi APC. Kemudian sel T yang teraktivasi ini akan dicek kecocokan HLA-nya dengan pasien yang mau di treatment. Gitu. Jadi kalau kita lihat dan kemudian setelah HLA-nya dipastikan cocok, maka dia yang akan diperbanyak dan disimpan dalam bentuk biobank. Next slide. Jadi tadi jadi sel ini gitu ya, sel T yang sudah teraktivasi spesifik mengenali EBV positif sel gitu setelah dicek kecocokannya dengan HLA gitu dari pasiennya, maka dia akan diberikan dan karena dia HLA-nya cocok dan sel T-nya sudah teraktivasi, maka dia akan mengeliminasi sel B yang ada di pasiennya gitu sehingga sel-sel yang tidak EBV positif itu tidak akan dieliminasi. Jadi hanya sel-sel yang EBV positif yang ada di pasiennya yang akan dieliminasi oleh sel T yang diberikan ini. Yang jadi memang mekanismenya agak sedikit complicated gitu ya karena dari produknya sendiri dia mau mentarget ke sel yang EBV positif saja. Nah, itu contoh yang sel terapi. Kemudian next slide. Ini adalah yang tissue engineered medicine. Jadi dia lebih bentuknya ke arah jaringan gitu ya. Jadi meregenerasi jaringan kita. Ini ada dua contoh. Yang pertama adalah holoclar. Next slide. Langsung aja Mas. Ya, di sini holoclar itu dia mengambil corneal epithelial cell dari manusia, autologous sistemnya, jadi dari orang itu sendiri diambil sel epitel dari kornea yang masih sehat, kemudian diekstrak selnya menjadi primary cell culture gitu ya, kemudian ditumbuhkan gitu, diperbanyak gitu, dan kemudian dikembalikan lagi ke pasiennya untuk memperbaiki kondisi korneanya gitu. Kemudian ada satu produk lagi, ini adalah Spherox untuk memperbaiki chondrocyte gitu yang ada di tulang. Jadi diambil sel gitu ya secara ex vivo, chondrocyte-nya dikeluarkan, kemudian diekspand. Diekspandnya dikultur dalam bentuk 3 dimensi sehingga dia mampu membentuk yang namanya spheroid. Spheroid ini kemudian yang akan disuntikkan kembali ke pasiennya supaya dia membentuk jaringan yang diinginkan gitu ya. Jadi kayak memberikan implan gitu ya, tetapi dari sel yang sudah kita kultur secara 3 dimensi gitu. Next slide. Nah, jadi ketika dia dikultur secara in vitro, itu gambarannya seperti yang di kiri gitu ya, dia akan membentuk kumpulan sel, makanya disebut dengan sferoid, dan nanti setelah diinjeksikan, sel-sel ini akan membelah dan kemudian menutup daerah yang ditargetkan seperti di gambar yang kanan. Gitu ya, jadi mengobati kondisi um, apa yang ingin ditargetkan. Nah, mungkin dari saya itu saja untuk mendapatkan gambarannya. Jadi, entah dia bentuknya terapi gen atau sel terapi atau tissue engineered medicine, tentunya kita harus melihat bagaimana mekanisme kerja yang ingin ditargetkan ini bisa berjalan nantinya di pasien, kemudian bagaimana menghantarkannya, apakah dengan viral vector atau non-viral vector gitu ya. Tentunya tanpa melupakan konsep utama untuk setiap terapi, yaitu dari segi kualitas, keamanan, dan efikasi. Itu saja dari saya, Ibu Bapak. Terima kasih atas perhatiannya.
Ya, terima kasih Bu Anita untuk paparannya. Bagaimana pagi-pagi sarapannya materi Bu Anita ini mantap ya teman-teman. Ya, Ibu tadi sudah menyampaikan overview tentang ATMP ya. Mungkin sebelum kita diskusi, kita lanjutkan dulu materi kedua dari Prof. Amin tentang peran akademisi dalam pengembangan itu. Kan Prof, langsung saja. Ya.
Baik, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Salam sehat, salam sejahtera buat kita semuanya. Terima kasih kita pagi ini sudah diberikan kuliah 3 SKS dari Bu Anita. Ya, luar biasa. Jadi meringankan tugas saya. Ada beberapa slide juga saya membahas lagi ATMP ini. Saya dapat tugas yang agak susah juga sebetulnya ini. Peran akademia dalam pengembangan ATMP ini, ya, khususnya tentu di Indonesia. Slide berikutnya. Slide berikutnya malah habis.
Lagi ya?
Terus saya share screen dari komputer saya.
Bisa, Mas? Atau mungkin
Oh, sudah, sudah. Oke. Oh, karena bukan
PDF.
PPT. Saya rekam dari saya, jadi dari laptop saya aja. Mohon izin Ibu dan Mas sekalian, saya presentasi dari meja saya aja. Enggak apa-apa. Biar saya dekat sama Bu Cynthia. Nggak apa-apa. Nah, ini kan kelihatan. Jadi saya dekat minuman makanan. Baik, judulnya ini peran akademia dalam pengembangan ATMP. Juga nggak mau diklik ini. Oke. Tentang ATMP, jadi saya nggak mengulang lagi apa yang sudah disampaikan oleh Bu Anita tadi. Intinya bahwa kita sepakati ini untuk menyamakan persepsinya aja bahwa ATMP itu nggak apa-apa, ya lebih enak ini. Ya terdiri dari tiga jenis utamanya. Eh, Istilahnya adalah untuk karakteristiknya. Jadi ada gen terapi, ada sel terapi, dan ada tissue engineering. Tapi sebetulnya ATMP ini dibuat untuk bisa memenuhi apa yang sudah ada sekarang dirasa belum bisa mengobati beberapa jenis penyakit ya. Jadi diharapkan dengan adanya ATMP ini beberapa penyakit yang tidak bisa ditangani dengan obat-obat farmaseutika yang sebelumnya ini bisa diupayakan dan ujungnya sebetulnya yang diarahkan adalah personalized medicine atau precision medicine. Ya. Nah, ini bukan definisi sih, tapi untuk menyamakan persepsi sebetulnya ATMP itu apa aja. Ya. Jadi sebetulnya ATMP itu dianggap sebagai pharmaceutical, jadi sebagai obat. Ya. Tapi obatnya agak berbeda dengan obat yang sudah ada sekarang, yaitu obat yang didasarkan pada modifikasi genom dari pasien ataukah penambahan atau rekomendasi dari asam nukleat dari gennya dari pasien tersebut, kemudian juga manipulasi dari sel-sel yang diambil dari subjek, ya. Nah, kemudian ada juga karena kita tahu kan ada sel yang diambil dari orang kemudian diberikan kepada orang lain, misalnya donor darah. Tapi kalau yang ini ditujukan namanya non homologous, artinya digunakan untuk keperluan jaringan lain. Ya. Nah, kemudian juga ada tisu yang direkayasa. Secara singkat tadi sudah disampaikan bagaimana ATMP itu dibentuk, tapi intinya ada dua teknologi ya. Yang satu adalah didasarkan pada gene teknologi, artinya bisa dipotong, dikurangi, ditambah, diubah, ya. Atau didasarkan pada jaringan atau sel dari manusia yang kemudian dimodifikasi. Nah, tujuannya sih semuanya sama ya, untuk memperbaiki DNA dari pasien, untuk menyembuhkan gangguan-gangguan yang ada karena disebabkan karena ada kelainan gen, ya. Kemudian juga untuk mengganti sel-sel yang atau jaringan yang rusak. Nah, itu tujuannya sih semuanya ke arah sana, tapi sifatnya lebih banyak ke individual. Nah, secara lebih rinci lagi kita sebenarnya bisa kelompokkan ATMP itu menjadi yang sekali lagi didasarkan pada terapi gen atau didasarkan pada terapi sel. Ya. Nah, tapi semuanya itu dimasukkan dalam ATMP. Nah, yang ingin saya tekankan bahwa ATMP itu biasanya diawali dengan adanya selalu ya, ada donor, ada resipien. Resipiennya itu bisa orang yang sama, bisa orang yang berbeda. Ya. Nah, kemudian juga ada teknologi yang dimasukkan yaitu rekombinan gene teknologi. Ya, jadi itu alternatifnya. Baik, saya akan lanjutkan. Saat ini saya mulai masuk yang ke arah akademiknya nih. Saat ini penerapan dari ATMP sudah sangat banyak, tapi yang paling banyak diterapkan adalah untuk hematological cancer dan limfoma. Itu yang saat ini paling banyak dipakai. Ya. Tapi dasarnya adalah tadi yang sudah disampaikan oleh pembicara sebelumnya, dasarnya adalah gene therapy, ya, somatic cell therapy sedikit. Kemudian genetic disorders ini juga sudah banyak diterapkan. Yang berikutnya adalah endokrin, nutrition, dan itu misalnya pada pasien-pasien yang DM dan sebagainya. Kemudian ada beberapa penerapan sedikit lagi di kardiovaskuler. Dan tadi satu contoh yang disampaikan oleh pembicara sebelumnya adalah penerapan di mata. Jadi memang mungkin ke depannya kita tidak perlu lagi donor kornea, ya, karena kornea itu sudah bisa dibuat, dikultur, bisa kemudian ditanam kembali. Baik. Nah, tadi sudah banyak contohnya, tapi kalau kita lihat sebenarnya ATMPD bukan baru ya. Buat kita baru barangkali ya. Tapi di luar sana, di EU misalnya, itu 2009 sudah ada produk yang di-approve oleh mereka, yaitu Condro Select. Ini bacanya Condro, kalau Condro itu mungkin ayam goreng, ya. Ayam goreng Bu Condro itu ya. Tapi ini Condro Select. Kemudian ada Glibera di 2012 ya. Di US baru belakangan ya 2010, walaupun registrasinya 2009 juga sih sebetulnya. Yaitu somatic cell therapy untuk mengobati kanker prostat. Nah, bagaimana peran akademia? Ya, sebetulnya dalam pengembangan jadi penelitian dan pengembangan ATMP ini peran akademia ini sangat banyak ya, mulai dari R&D, kemudian uji klinik. Uji klinik biasanya tidak bisa lepas dari akademia. Kemudian kolaborasi dengan industri ya. Nah, ini juga yang penting evidence based policy recommendation. Jadi kita menghindari policy driven evidence development. Ya, jadi jangan dibalik. Ya, mestinya policy recommendation itu evidence based dan semua dilakukan di lingkungan akademia. Kemudian tentu edukasi, nanti saya uraikan satu persatu ini beberapa di antaranya. Dan juga tentu kolaborasi secara global. Clinical trial misalnya. Sampai saat ini saya baca laporannya sampai dengan Juni 2024 nih. Ya, sudah ada 900 lebih clinical trial ATMP yang dikerjakan oleh ratusan ya 300 lebih, hampir 400 developer. Jadi sangat banyak pemainnya sebetulnya. Kemudian yang saat ini sudah dalam pengembangan ke arah yang lebih advance itu sekitar 600-an, tapi 60% dari clinical trial itu adalah untuk gene therapy. Jadi sama seperti yang grafik yang sebelumnya, penerapannya memang lebih banyak di gene therapy. Pengembangan yang sekarang sedang itu diarahkan untuk hematological cancer dan limfoma. Nah, kalau kita lihat di graf sebelah kanan ini, kita lihat bahwa clinical trial yang paling banyak saat ini adalah fase 1 yang menuju fase 2 dan juga fase 2 itu sendiri. Ya. Yang di fase 3 masih relatif sedikit, apalagi fase 4. Jadi ini fase 4 ini kan biasanya post-marketing ya. Jadi kita lihat sebagian besar pengembangan ATMP ini masih di fase 2 dan fase 3. Kemudian kalau yang kita lihat lagi apa yang sudah dikembangkan, yang sudah mendapatkan license itu yang biru ini sebelah kanan, ya. Ada sekitar 50-an, tapi sebagian besar adalah masih dalam pengembangan, ya, sekitar 600-an tadi sudah saya sebutkan. 600 lebih itu masih dalam pengembangan. Yang sudah pre-registrasi dan sebagainya ada 1 dan 2. Tapi ingin saya tunjukkan di sini ada yang biru, satu pojok paling bawah nih, ya. Ada yang withdraw, artinya ini sebenarnya ada contohnya yang lain lagi. Yang sudah dikembangkan itu kemudian aborted karena masalah teknologi, masalah pemasaran, masalah harga dan sebagainya. Nah, itu yang mungkin juga perlu mendapat perhatian kita semuanya. Dari distribusi geografiknya saat ini yang paling berkembang memang di US, ya, Kanada, kemudian di negara-negara Eropa, dan juga kita lihat India dan Cina sudah mulai ada. Indonesia belum ada yang mampir nih. Nggak muncul. Ini masalah mata atau masalah komputer? Ini masalah ATMP kali. Ya. Nah, di Zoom ada. Salah layarnya kalau gitu. Ya, oke, saya lewatin aja. Nah, jadi dari bahasan tadi sebetulnya kita melihat bahwa akademia itu sangat erat kaitannya dengan ekosistem inovasi. Ya. Nah, ekosistem inovasi untuk ATMP ini melibatkan beberapa institusi, ya, utamanya karena akademia sebagian besar adalah warga universitas. Ya. Tapi ada juga yang tidak terkait langsung dengan universitas, tapi dengan research institute. Hal yang sangat mendukung adalah yang kita lihat juga dari sejarah keberhasilan pengembangan produk-produk ini, itu dikembangkannya bukan oleh perusahaan farmasi yang besar-besar, tapi oleh startups, ya. Itu yang memulai. Nah, baru setelah sukses, baru ditangkap oleh perusahaan farmasi yang besar-besar. Ya. Nah, itu semuanya sangat dipengaruhi oleh cikal bakalnya oleh para akademia tentunya. Ya. Nah, kemudian bagaimana akademia itu kolaborasi dengan industri, ya. Kita lihat ada dua hal yang mempengaruhi. Pertama dari sudut kualitasnya, yang kedua adalah standpoint terkait dengan clinical dan non clinical. Ya, kalau dari quality standpoint-nya itu ini yang dialami juga oleh peneliti Indonesia, ya. Biasanya peneliti-peneliti itu sangat semangat, tapi pada awalnya biasanya sangat soliter, kerjanya sendiri, kalau bisa jangan ada orang lain lihat, ditutupin semuanya, nanti begitu udah jadi baru ditawarkan ke industri. Nah, padahal prosesnya barangkali tidak sesuai dengan proses industri. Nah, jadi ini yang harus kita sampaikan juga kepada teman-teman akademia yang mau mengembangkan, ya. Karena harus ada harmonisasi antara apa yang dilakukan di akademia dengan yang di industri. Kemudian kalau yang non clinical sama clinical itu pasti sudah terkait dengan bagaimana itu akan diaplikasikan, kemudian terkait dengan patient safety dan sebagainya, ya. Sampai kepada nanti kalau ada clinical trial 2, 3 dan seterusnya, tentu kita bicara tentang dosis, tentang keamanan dan sebagainya. Ya. Nah, faktor lain yang dihadapi oleh para akademia ini adalah juga terkait dengan yang sekarang sedang kita bahas nih, regulatory dan ethical landscape. Ya, hari ini mungkin kita nggak akan bahas mengenai ethical ya, tapi untuk akademia pasti kita tidak bisa lepas dari LC ya, ethical, legal, dan social implication. Karena penerapan terapi sel, terapi gen, stem cell dan sebagainya itu sangat erat kaitannya dengan ethical. Saya kira di Surabaya nanti minggu depan atau 2 minggu lagi akan ada pertemuan di salah satu rumah sakit ya, Dr. Heri ya. Saya diundang ke sana untuk pengembangan etik penelitian terkait dengan terapi-terapi ATMP ini. Oke, nah terkait dengan regulasi ini contoh aja nih. Bagaimana akademia itu seringkali dihadapkan pada masalah regulasi yang ya mungkin berbeda satu sama lain. Kita ambil contoh misalnya, kalau kita berkiblat ke EU, maka seperti tadi kita bahas dari awal, ATMP itu dikelompokkan menjadi ada 4 ya. Ada yang GTMP ya, itu gene therapy, medicinal product, kemudian ada yang somatic cell therapy, kemudian ada yang tissue engine product, dan ada lagi yang combine ya, si ATMP. Itu mereka bagi 4. Tapi kalau di US, mereka hanya bagi 2, gene therapy dan cell therapy. Ya, sudah selesai. Nah, tentu aturan-aturannya akan berbeda juga. Ya. Nah, ini para akademisi juga mesti mencermati hal ini, ya, karena mungkin pertimbangannya adalah nanti di mana kita akan kerjasama, di mana kita akan memasarkan produk dan sebagainya. Berikutnya masalah edukasi, ya, ini peran juga peran dari akademia. Kita harus memberikan edukasi tentu satu ke mahasiswa, ada pastilah gitu ya, karena ilmuwan akademia itu tidak lepas dari mahasiswa baik S1, S2, S3. Tapi juga kita punya kewajiban untuk memberikan edukasi kepada publik, ya, karena publik itu seringkali juga ada over ekspektasi atau juga yang mereka nggak tahu kalau teknologi itu sudah ada. Yang ketiga adalah tentu teman-teman di bisnis juga gitu harus kita berikan tanda petik edukasi, dan jangan lupa government ya, agar kebijakan yang diambil itu betul-betul evidence based, maka kita harus rajin-rajin memberikan edukasi kepada pihak government tentunya. Nah, tantangan apa yang dihadapi oleh akademia dalam pengembangan ATMP? Ya, yang pertama masalah teknis sudah pasti lah gitu ya, tapi ini tantangan yang juga menjadi opportunity sih sebetulnya. Ya, kita lihat tadi dari kuliah 3 SKS tadi yang pertama itu sudah kelihatan sekali teknologinya, beberapa barangkali belum ada di Indonesia barangkali ya, atau masih terkait dengan masalah etik dan sebagainya. Nah, tapi tantangan yang berikutnya adalah soal funding ya, karena penelitian ATMP ini, ATMP, stem cell dan sebagainya itu luar biasa ya. Belum lagi funding untuk pengobatannya. Tadi salah satu contoh yang disebutkan adalah Kartisel misalnya yang sudah ada di beberapa Indonesia. Saya nggak tahu apakah sudah di-approve oleh Badan POM apa enggak. Belum ya. Karena satu kali treatment aja 4-5 miliar. Nah, kemudian tantangan yang berikut adalah dari regulator. Ya. Jadi biasanya sangat kompleks. Di sini ada Dr. Wiwi di sini, kita bikin aturannya sama-sama Kemkes dengan Badan POM. Targetnya September yang tahun lalu udah jadi, tapi sekarang sampai September tahun ini juga belum selesai juga. Ya, jadi karena begitu kompleksnya, jadi kita ingin aturan itu memang ada, tapi juga artinya mendukung, tapi juga memastikan bahwa teknologi ini bermanfaat, tapi juga aman untuk pasien. Yang berikutnya adalah public perception ya, dan juga acceptance. Ini juga seringkali menjadi tantangan. Pertanyaan apakah dipakai media dari hewan, sumbernya dari hewan apa enggak, ya, selnya dari negara Indonesia sendiri apa atau dari luar, ya masalah-masalah gitu seringkali menjadi pertanyaan. Baik, tapi tentunya dari segala tantangan, kesulitan dan sebagainya ada success stories. Tadi beberapa sudah disampaikan, ini daftar ini bukan untuk dihafalin ya ibu-ibu, bapak-bapak, hanya mungkin dari belakang kelihatan tapi tidak terbaca. Ya. Intinya adalah sudah ada produk-produk ini yang ATMP ya, berdasarkan gene terapi, kemudian ada yang berdasarkan somatic cell therapy, medicinal product, itu sudah ada yang dipasarkan, kemudian dari tissue engineer medical product juga sudah ada yang dipasarkan saat ini. Ya. Ini data di tahun 2021. Baik, sebagai kesimpulan, yang pertama kita semua sepakat bahwa ATMP ini adalah termasuk masa depan dari pengobatan. Ya, masa depan farmaseutical ini adalah ATMP karena sangat promising untuk pengobatan ya, baik melalui genetic engineering, kemudian kemajuan teknologi dalam biomanufacturing, dan juga terkait dengan personalized medicine. Kemudian kita juga sepakat bahwa akademia ini memiliki peran yang sangat penting ya untuk mulai dari R&D-nya, kemudian membangun kolaborasi baik dengan industri, dengan ya, pihak-pihak lain. Ya, jadi ini sekaligus mendorong semua akademia untuk tidak berputus asa nih, ya, karena tantangannya memang besar, tapi peluangnya juga sangat besar. Demikian yang dapat saya sampaikan, Ibu dan Bapak sekalian, mudah-mudahan sedikit menambah wawasan. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Ya, terima kasih Prof. Amin untuk paparannya. Jadi 1 jam ini kita sudah dapat 6 SKS ya. Ya, mudah-mudahan dengan pemaparan kedua dari Prof. Amin semakin jelas ya, bukan semakin pusing ya. Kita sekarang akan beralih ke sesi tanya jawab. Pastikan banyak ya, wah itu kan sudah banyak mengacungkan tangan saya lihat di sini ya. Saya mohon bersabar. Baik, untuk sesi tanya jawab kami berikan kesempatan terlebih dahulu kepada para peserta yang hadir secara luring. Nanti mohon bantuan dari panitia jika ada yang bertanya melalui daring ya. Mungkin nanti bisa ditampilkan pertanyaannya untuk bisa dijawab. Baik, untuk sesi pertama pertanyaannya silakan sebutkan nama, dari mana, dan diajukan kepada siapa.
Ya, baik. Terima kasih Profesor Amin dan Dr. Anita. Nama saya Mayerni. Saya dari Badan POM dan saya di bagian Direktorat Registrasi Obat bertugas sebagai evaluator obat. Izin Prof, memang tadi dari pemaparan itu tadi disampaikan bahwa ATMP ini merupakan hal yang baru dan ini sangat menarik sekali ya Prof ya. Nah, kemudian artinya kita melakukan manipulasi kepada gen. Nah, kemudian ketika tadi paparan muncul di benak saya, ini saya sebagai evaluator artinya saya bertugas ketika ada satu produk ATMP kita harus memastikan apakah produk itu nanti efektif, aman, dan bermutu. Nah, yang ingin saya tanyakan ada dua. Bagaimana memastikan bahwa modifikasi seluler yang dilakukan tidak menyebabkan proliferasi sel yang tidak terkontrol atau perubahan sifat biologis yang tidak diinginkan. Nah, misalnya pada tisu engineered dan pada somatic cell therapy. Nah, itu bagaimana? Apakah ada resiko terjadinya pembentukan tumor seperti itu, Prof? Kemudian yang kedua, tadi dari paparan Dr. Anita disampaikan bahwa nanti ada prosesnya seperti apa? Seperti nanti diambil gennya, kemudian nanti ditransfer. Nah, apakah ada risiko terjadinya kontaminasi dengan patogen atau agen infeksius dalam proses produksi terapi berbasis sel? Nah, sebagai evaluator obat, kira-kira apa yang harus kami perhatikan, Prof. Dan Bu Anita? Terima kasih.
Baik, Ibu, terima kasih atas pertanyaannya. Tadi ya modifikasi yang dihasilkan gitu ya, perlu dipastikan apakah ada efek yang tidak diinginkan gitu ya. Salah satunya tadi yang saya sempat singgung adalah off-target effect. Misalnya kita pakai gene editing gitu ya. Nah itu memang betul ada resiko bahwa dia mengedit di tempat yang lain misalnya. Itu maka menurut pendapat saya sebagai evaluator seharusnya dicek apakah em, eh, kalau itu ada industri yang memasukkan produknya gitu ya, bagaimana dia mengecek off target effect misalnya. Kalau dia hanya cek dari sequence yang ditarget, kan berarti yang tidak ditarget apa buktinya? Berarti kan itu yang perlu juga dianalisa dan bagaimana mereka mengevaluasinya gitu ya. Kemudian misalnya efek tumorigenicity gitu. Kita tahu kalau kita produksi AAV misalnya menggunakan HEC 293 itu ada resiko tumorigenicity dari residual host DNA ketika produksi. Bagaimana mereka mengecek tumorigenicity effect gitu kan? Itu ada kalau di Eropa mereka bisa menggunakan saya lupa ada nude mice atau apa gitu, tapi setahu saya ada pedomannya untuk mengecek resiko tumorigenicity menggunakan model hewan misalnya. Nah, itu sesuatu yang bisa dicek. Jadi, kalau kita tahu bagaimana produsen itu melakukan prosesnya, kemudian dengan dokumen yang disubmit, kita harus tahu mana yang harus dicek sebetulnya gitu ya, yang berkaitan dengan produknya. Dan itu nature-nya akan beda-beda tergantung tadi modifikasinya apa, kemudian bagaimana mereka melakukan validasi prosesnya. Termasuk diantaranya tadi yang terkait dengan kontaminasi, betul. Ketika dilakukan leukapheresis untuk mengambil sel T, bagaimana mereka memastikan sortir sel T-nya hanya sel T saja, itu satu. Dan tentunya ketika dilakukan sorting, kemudian transduksi dan ekspansi, di situ tidak ada kontaminasi misalnya. Kontaminasi ketika proses pengerjaan, maka itu yang harus dilihat dari segi resiko mutu. Setelah sel T-nya ditransduksi dengan CAR, bagaimana memastikan hanya yang membawa reseptor saja yang dikembalikan ke pasien atau dia harus ngecek berapa persen yang berhasil tertransduksi dan bagaimana mengeliminasi yang tidak ditransduksi. Dan itu kalau kita lihat dari aturannya, setelah sel itu keluar dari tubuh pasien maka harus dikerjakan di fasilitas dengan sertifikat CPOB atau GMP kan, dan di situ jadi mirip teknik aseptisnya dengan produksi produk secara aseptik secara umum, maka kontaminasi dan apa yang berkaitan dengan kualitas dan keamanan tentunya harus dikaji resikonya gitu. Kemudian kontaminasi dari donor misalnya. Salah satu contohnya kalau tidak salah, saya lupa yang Ferox atau yang Evalo, dia harus ngecek bahwa dari dari yang mendonorkan selnya itu tidak ada infeksi patogen tertentu, misalnya hepatitis B atau hepatitis C, dan itu kan sesuatu yang harus dimitigasi pada tahap awal. Mungkin itu yang bisa saya sharing itu untuk topik. Terima kasih.
Masuk. Baik, terima kasih. Pertanyaan menarik sekali. Saya nyambung dulu dengan tadi terkait dengan kontaminasi. Tentu donornya harus dipastikan mirip seperti kalau kita mau donor darah itu ada risiko TTI ya, transfusion transmitted infection. Kita mesti pastikan bahwa dia nggak ada HIV, nggak ada HCV, nggak ada sifilis dan sebagainya. Itu kalau dari kontaminasi, ya, kontaminasi di sumbernya. Tentu dalam proses berikutnya juga kita mesti pastikan tidak terjadi kontaminasi bakterial, jamur dan sebagainya. Itu yang menyebabkan kita mesti punya fasilitas yang memenuhi persyaratan. Kemudian pertanyaannya apakah tidak ada risiko perubahan-perubahan sampai ke tumorogenesis dan sebagainya ya. Tentunya sebelum diberikan kepada manusia itu kita harus melakukan penelitian ya untuk melihat efek jangka pendek, jangka panjangnya, efek samping atau efek simpang, termasuk memastikan bahwa tidak terjadi perubahan yang tidak kita inginkan. Salah satu misalnya kita mesti memastikan target organ yang akan diberikan itu sama nggak dengan yang akan diberikan donor. Artinya gini, kan kita tahu ada mesoderm, ada ektoderm, ada endoderm ya. Nah jadi kita hindari stem cell yang berasal dari misalnya dari endoderm diberikan ke untuk yang di jaringan ektoderm, itu memang dihindari karena tetap aja ada risiko ya. Saya nggak akan bilang bahwa tidak ada risiko. Ya risiko tumorogenik itu ada untuk jadi teratoma dan sebagainya itu mungkin saja ada. Tapi itu biasanya setelah dilakukan berbagai penyesuaian itu bisa diperkecil. Tentu pertimbangannya adalah kita mesti lihat risk and benefit ratio-nya. Ya, risikonya ada, tapi seberapa besar? Ya, tapi kalau benefitnya dianggap lebih besar, itu yang kita ambil. Terima kasih.
Ya, terima kasih Prof. Mbak Anita. Mbak Mayerni, apakah sudah terjawab? Atau mau ada lanjutan pertanyaan?
Iya.
Di luar.
Iya, tadi yang pertamanya itu bagaimana kita memastikan dia proliferasinya tidak terkontrol itu kira-kira bagaimana ya? Tadi kan dari penjelasan Prof dan Dr. Anita menyatakan bahwa kalau untuk tumor mungkin bisa ke hewan seperti itu ya, Prof. Kalau untuk kita memastikan dia itu tidak akan terjadi perubahan proliferasi sel yang tidak terkontrol atau perubahan sifat biologis yang tidak yang kita harapkan itu kita kira-kira apa ya? Karena tumor itu kan perlu waktu yang panjang ya, Prof. Kira-kira bagaimana?
Mungkin kalau untuk saya jangka panjang saya sendiri kurang tahu gitu ya Bu ya, tapi ada nggak dari produsen itu kemudian mencoba mengkultur sel hasil modifikasi itu selama berapa pasase, apakah ada perubahan fenotipe, genotipe misalnya gitu ya. Kalau misalnya kayak tadi CAR-T kan sel T-nya diambil lalu dimodifikasi, itu kan secara in vitro juga bisa dikultur dalam jangka panjang atau pada kondisi tertentu gitu ya, sebagai uji pendahuluan mungkin ya untuk memastikan sampai berapa pasase dia mempertahankan sifatnya atau ada yang berubah atau tidak. Saya nggak tahu apakah mungkin ini kemudian ada pengujian yang bisa paling tidak in vivo gitu ya, tetapi masih di hewan. Saya belum pernah baca sih, tetapi secara logika mungkin masih bisa ke arah situ gitu. Mungkin Prof. Amin lebih banyak pengalaman untuk yang uji preklinis gitu ya. Tapi gambaran saya sih seperti itu. Kalau ada bukti secara in vitronya, kemudian dia bisa dibuktikan secara in vivo, mungkin itu salah satu cara yang bisa dilihat.
Satu contoh adalah tadi kan disinggung salah satu kan teknologinya kan menggunakan adenovirus. Untuk vaksin juga gitu ya. Vaksin COVID kemarin juga kan ada yang pakai. Itu juga ada dua jenis, ada ya virus yang replicating sama yang non-replicating. Yang tentu yang lebih disukai adalah yang non-replicating. Virusnya hidup tapi tidak replikasi. Nah, yang replicating ini juga di program sedemikian rupa sehingga ada untungnya dia replikasi sehingga kita nggak usah kasih dosis tinggi-tinggi, kita kasih dosis 10 aja nanti dia akan replikasi sendiri menjadi 100 misalnya, tapi begitu mencapai 100 dia akan berhenti. Jadi itu yang harus dikendalikan. Nah, dengan sel-sel cancer juga demikian, ya kita bisa program sebetulnya sampai berapa kali dia replikasi sel-sel yang kita masukkan. Walaupun memang harus diakui ya ada aja yang nyeleneh gitu ya, satu yang 100 90 sel itu bisa kita kendalikan, nggak tunggu-tunggu lagi, tapi yang 10 barangkali ya merupakan ya nyeleneh gitu ya, itu bisa mungkin terjadi sih. Waktu kita seleksi mungkin ada yang lolos. Sama juga seperti kalau kita memberikan vaksin yang hidupnya dilemahkan, kita nggak bisa jamin bahwa 100% itu dilemahkan. Mungkin 1 atau 2 virus itu masih virulen, dan itu bisa kembali jadi virulen. Terima kasih.
Terima kasih.
Baik, terima kasih Prof. Dr. Anita.
Terima kasih. Jadi Dr. Heri mau tanya.
Baik, terima kasih. Pertanyaan pertama sudah. Untuk berikutnya tadi saya lihat sepertinya banyak ya yang ngacungkan tangan. Nah, Dr. Heri silakan. Dok.
Terima kasih, saya Heri Suroto dari Bank Jaringan dan Sel RSUD Dr. Sutomo. Terima kasih kami mengikuti dengan seksama apa yang disampaikan Ibu Anita dan juga Prof. Amin. Ya. Kalau kami mencoba untuk memahami bagaimana sel, khususnya dalam kaitannya dengan ATMP, jadi sel itu dia bisa diambil, kemudian bisa dilakukan sesuatu, apakah itu dengan gene terapi ataukah dengan sel terapi, kemudian baru diimplantasi. Nah, sekarang pada saat tissue engineering produk tadi Ibu Anita menyampaikan, yang saya lihat adalah masih berputar sekitar sel. Ya. Nah, kami di Bank Jaringan itu, sebagaimana kemarin juga kami sudah tanyakan, malah kita berusaha untuk membuang selnya. Tetapi dengan catatan, bagaimana jaringan yang kita produksi, apakah itu jaringan tulang, apakah jaringan tulang rawan, apakah jaringan saraf, itu mimicking dengan jaringan aslinya. Sehingga kalau kami produksi jaringan manusia, jadi kami di Bank Jaringan itu banyak spare part-nya Ibu, ya tulang, ya tendon, ya saraf, ya fasia. Bahkan kalau orang sunatan kulitnya pun yang terbuang masih bisa kita pungut. Jadi kita itu tidak ada yang terbuang. Nah, pertanyaan saya Ibu, pada saat kita mencoba untuk mimicking, menyerupai dengan jaringan yang ada, ya, bagaimana bisa ada satu komunikasi antara sel dengan jaringan. Kalau tadi saya melihat CAR T-cell itu di treatment sehingga ada SCFV itu dia bisa connect dengan CD19 ya. Nah, sekarang pertanyaan saya bagaimana kalau kita implantasi jaringan yang sudah kita proses, ya, itu bisa sel itu melekat karena itu merupakan suatu yang kunci. Sehingga akhirnya pada saat sel yang melekat tadi, dia bisa berproliferasi, bahkan dia bisa berdiferensiasi menjadi sel tulang, bahkan memproduksi jaringan tulang. Ya. Itu pertanyaan pada Ibu Anita. Kemudian kepada Prof. Amin, izin Prof. Kalau kita melihat bahwa sel dengan sel itu ada satu komunikasi, tentunya juga sel dengan jaringan ada satu komunikasi. Ya. Kalau di komunitas ini juga ada komunikasi. Tadi Prof menyampaikan ada akademisi, ada bisnis, ada klinisi. Ya, saya sebenarnya terus terang bukan saintis, tetapi saya klinisi. Jadi dalam klinisi ini sering saya melihat tulang itu hilang oleh karena trauma, oleh karena infeksi. Bagaimana kita mengganti jaringan trauma itu, ya. Jaringan tulang yang hilang tadi. Ya. Nah, sehingga yang kami lakukan di tissue bank itu kita bagaimana bisa menyediakan provide jaringan-jaringan yang biasanya hilang, terutama trauma. Ya. Kalau trauma itu kan selnya masih sehat, tidak ada penyakit, sehingga tentunya pada saat ada penggantian jaringan itu akan bisa diharapkan melekat. Nah, cuma memang di dalam implementasinya komunikasi itu masih kurang terjalin dengan baik, Prof. Jadi mungkin kita kalau di Kementerian Kesehatan ada komite sel punca, ada komite transplant organ dan jaringan, ada nama jaringan tetapi sering kita itu merasa orang jaringan itu kurang mendapatkan perhatian. Padahal transplantasi jaringan itu angkanya luar biasa, Prof. Sejak tahun 90 sampai sekarang kami ada catatannya setiap tahunnya. Nah, cuma sekarang bagaimana memungkinkankah komunikasi itu di tingkat jaring, artinya memungkinkankah dalam komunikasi ini sehingga ada suatu produk jaringan. Sebetulnya kami sudah memproduksi jaringan, sudah melakukan transplantasi jaringan, bahkan jaringan itu dari Aceh sampai Papua kita distribusikan. Cuma memang pada saat mulai adanya BPJS itu rumah sakit meminta NII. Nah, NII itu yang kemarin juga masih belum terakomodasi. Padahal secara kebutuhan itu sudah sangat banyak orang memerlukan tulang, orang memerlukan tulang rawan. Ya. Dan itu sebetulnya kita ada produk itu. Nah sekarang mohon izin kalau seandainya dalam komunitas ini bagaimana kita mengimplementasi akan kebutuhan pasien, saya sebagai seorang klinisi, itu bisa menjadi satu produk yang kalau kemarin mungkin juga bukan produk ATMP, tetapi sebetulnya secara praktek itu banyak terjadi, banyak kita lakukan transplantasi jaringan. Terima kasih, mohon arahan.
Terima kasih atas pertanyaannya. Jadi memang tadi yang dikategorikan dalam ATMP untuk yang tissue engineer itu memang dia berangkat dari sel gitu ya dok ya. Tapi dari sel kemudian bagaimana dia dimodifikasi supaya bisa membentuk jaringan. Nah ini tentunya tadi kalau dari pertanyaan yang saya tangkap ini kan bagaimana komunikasi antara sel ketika membentuk jaringan atau sel dengan jaringan gitu ya, tentunya di situ harus dipelajari dulu bagaimana kayak tadi misalnya yang chondrocyte gitu ya, ketika dia dikultur dia diambil sel chondrocyte kemudian diseleksi lalu dikultur 3D, nah itu di situ ada ketentuannya yang bisa dikembalikan lagi, misalnya dia harus mengekspresikan protein apa-apa dan tidak ada ekspresi berlebih dari protein apa-apa karena dari situ kita memastikan bahwa mekanisme yang mau kita tuju tercapai kalau dia akan akan membentuk jaringan yang ingin kita bentuk di dalam tubuh gitu misalnya. Jadi memang harus dipelajari dulu tergantung itu sel apa dari jaringan apa dan bagaimana komunikasi antar selnya terjadi gitu. Misalnya antar sel epitel itu kan ada komunikasi antar sel sebetulnya dan ini yang bisa secara in vitro kita cek dulu dan bisa dijadikan menjadi parameter yang harus diuji sebelum hasil modifikasi itu dikembalikan lagi ke pasiennya. Mungkin secara gampangnya seperti itu, tapi saya lupa detailnya sebetulnya gitu ya. Tapi untuk yang spherox tadi ada sebetulnya kriterianya. Jadi spheroid yang siap untuk dikembalikan lagi ke pasiennya, dia harus mengekspresikan protein-protein tertentu gitu sebagai marker dia sebagai kondrosit dan nanti kondrosit yang memang akan berfungsi di dalam tubuh. Begitu.
Kalau Ibu menyebut spheroid tadi, saya melihat no suture, no fibrin glue. Cuma pertanyaan saya, bagaimana dia bisa melekat tanpa ada satu terlepas lagi?
Ya.
Saya terus terang belum bisa paham itu.
Iya.
Terima kasih.
Nah, itu juga itu salah satu yang ketika dilakukan kultur 3 dimensi, itu dia memerlukan beberapa suplemen dari luar gitu. Dia bisa berupa matriks gitu ya, yang memungkinkan dia membentuk 3D culture gitu. Jadi nggak hanya sel aja sebetulnya. Jadi ada beberapa protein-protein yang merupakan protein matriks ekstraseluler yang bisa ditambahkan ketika kultur sehingga dia membentuk seperti kultur 3 dimensi gitu. Saya memang nggak hafal detailnya gitu ya, tetapi pernah mengerjakan dengan 3D culture bukan untuk chondrocyte gitu ya, tetapi untuk sel epitel gitu, itu suplemennya banyak sekali gitu, seperti misalnya integrin atau laminin gitu, yang merupakan protein-protein matriks ekstraseluler yang membuat dia mampu tumbuh secara 3 dimensi. Nah, ini berkaitan dengan tadi presentasinya Prof. Amin, funding. Karena protein-protein itu biasanya harganya mahal-mahal sekali gitu ya. Dan kombinasi dok biasanya, jadi nggak cukup hanya satu protein, tapi dia bisa berupa kombinasi beberapa protein matriks ekstrasel gitu.
Saya tambahkan, Dr. Hedi. Jadi terkait dengan transplantasi, tadi kan untuk kalau trauma ada jaringan atau tulang yang hilang, itu kan diganti. Dulu sejak berapa tahun yang lalu ya, kita kalau misalnya tulangnya ada yang hilang, kan suka diganti dengan tulang sapi malah ya, yang diradiasi. Tapi ini yang ini tidak termasuk ATMP, karena yang tidak termasuk ATMP itu artinya kalau misalnya jaringan tulang yang memang kita pakai untuk tulang saja, tanpa ada modifikasi apapun, apalagi kalau jaringan tulang itu sudah tidak mengandung sel lagi, itu udah bukan ATMP lagi. Ya. Tapi kalau kemudian jaringan tulang itu kita modifikasi, misalnya kemudian kita olah selnya, diambil, kemudian dimodifikasi, ya. Apalagi kalau mau dipakai non homolog, artinya kita ambil stem cell, sel lah ya. Kemudian kita dari satu jaringan A, tapi akan dipakai di jaringan B. Nah, itu baru masuk di kriteria ATMP. Tapi kalau yang rutin sehari-hari dikerjakan tanpa modifikasi, itu nggak masuk di ATMP. Tapi saya sangat setuju bahwa kita perlu punya bank jaringan, itu perlunya Rija Selindo ya, karena kita memastikan misalnya ada satu center, satu daerah membutuhkan jaringan tulang misalnya atau jaringan kulit, nah itu misalnya sudah ada di tempat lain, itu tinggal dikirim. Tapi tentu persyaratan bank jaringan itu ada sendiri, itu terpisah. Saya kira sudah ada Kemenkesnya ya. Tapi itu di luar ATMP, itu nggak perlu masuk di ATMP. Ya, ATMP itu intinya kalau kita melakukan modifikasi sehingga sifatnya berubah atau akan dipakai di jaringan yang berbeda, nah itu baru kita lihat apakah ini masuk ATMP atau enggak. Ya. Terkait dengan tadi komunikasi antara sel, apakah transplant itu akan diikat oleh jaringan hostnya kan. Nah, itu ada beberapa pendekatan sih. Nah, antara lain adalah dengan memberikan satu mungkin scaffold, artinya yang menjembatani antara transplant dengan jaringan yang diberikan. Yang kedua memang memberikan semacam growth factors ya, untuk merangsang pertumbuhan sel di situ, ya. Atau ditambahkan stem cell memang atau ya stem cell atau produknya ya untuk merangsang pertumbuhan sel di situ. Biasanya pendekatannya sih seperti itu. Jadi kita nggak melihat lagi misalnya di tulangnya gimana selnya supaya tumbuh, tapi selama yang diberikan di situ jaringannya sama dengan sekelilingnya kan pertumbuhannya akan lebih bagus. Tapi kalau yang kita berikan tidak sama, misalnya tadi saya singgung sel yang berasal dari katakanlah mesoderm atau endoderm tapi ditransplantasikan di ektoderm, itu mungkin penolakannya akan lebih besar. Terima kasih.
Bagaimana Dok, sudah terjawab ya. Baik, terima kasih Prof. Amin, Bu Anita. Sepertinya masih banyak pertanyaan yang ingin disampaikan, tapi waktu juga yang membatasi kita. Tapi insya Allah ya, nanti kalau misalnya teman-teman ada pertanyaan bisa disampaikan melalui panitia, nanti kita bisa sampaikan kepada para narasumber. Baik, kami ucapkan terima kasih Prof. Dan juga Bu Anita untuk sharingnya pada hari ini. Mudah-mudahan bukan hanya menambah wawasan, tapi juga kita menambah jejaring ya. Tadi harapannya Dr. Heri juga ini perlu nih gitu ya, suatu komunikasi terkait dengan jaringan ini gitu yang sekarang belum ada gitu ya, meskipun itu bukan ATMP. Baik, mungkin itu saja sesi pagi ini. Kurang lebihnya mohon maaf. Assalamualaikum warahmatullahi wabarakatuh.
Terima kasih kepada Bapak Ibu narasumber dan moderator. Selanjutnya adalah penyerahan plakat penghargaan dari Badan POM kepada narasumber.
Ya, panggung. Iya. Baik, posisinya bisa di depan panggung. Mohon izin Bapak Ibu, plakat penghargaan akan diserahkan oleh Direktur Registrasi Obat Badan Pengawas Obat dan Makanan, Ibu Dr. Ya Christine Siagian, SSI Apoteker MSC. Kami mengundang Ibu Direktur untuk maju ke depan. Baik, penyerahan plakat penghargaan kepada narasumber pertama, ya, Dr. Renat APT Aluisia Anita Artarini, SSI MSC. Penyerahan plakat penghargaan kedua kepada narasumber Profesor PhD. Baik, boleh kita berikan tepuk tangan Bapak Ibu. Silakan untuk difoto bersama. Ya, boleh. Dengan moderator dipersilakan. Oke, boleh sekali lagi kita beri tepuk tangan. Terima kasih kepada Ibu Direktur, dan terima kasih kepada narasumber dan moderator yang telah berada di sesi workshop pada hari ini. Oke. Baiklah Bapak Ibu yang kami hormati, selanjutnya sesi workshop yang kedua akan membahas materi dengan judul Ensuring Quality Aspect of Advanced Therapy Medicinal Products yang akan disampaikan oleh narasumber dari Therapeutic Goods Administration TGA Australia.
Dilanjutkan dengan materi Strategy for Testing of Advanced Therapy Medicinal Products yang akan disampaikan oleh narasumber dari National Institute of Food Drug Safety Evaluation Korea Selatan.
Sesi workshop akan dipandu oleh moderator Ketua Tim Kerja Registrasi Obat Baru, Ibu Nani Sundari, SSI Apoteker and Biomed. Untuk itu kepada Ibu Nani Sundari, waktu dan tempat kami persilakan.
Assalamualaikum warahmatullahi wabarakatuh. Terima kasih kepada MC. Selamat pagi pada Prof. Amin, Prof. Muli, Dr. Siap, Dr. Hendy, dan para teman-teman semua. Kalau tadi sesi pertama nanti moderatornya ada narasumbernya. Kalau untuk sesi yang kedua ini, kedua narasumbernya akan hadir secara virtual. Jadi izinkan saya untuk membantu melihat pada sesi ini. Sepertinya untuk kedua narasumber kita sudah hadir. Mungkin bisa dipin ya Mas Mus, supaya lebih jelas untuk kedua narasumbernya. Iya, oke. Assalamualaikum warahmatullahi wabarakatuh. Good morning to Dr. Monica Srivastava from TGA. Good morning, Doctor. And good morning for Dr. Jin Wungkak from NIFDS Korea. Good morning, Doctor. Are you already joined with this session?
Good morning.
Okay, good morning, Doctor. Okay, good morning. I would like to open this session. Okay, good morning, ladies and gentlemen. Welcome to this second session for this today workshop. First of all, I would like to thank you to all the audience that already joined this workshop for the 3 days. And let me introduce myself. My name is Nani Sundari, and I will be serving as a moderator today for you, Dr. Monica and Dr. Jin Wukok. In this session, there are 2 speakers from National Regulatory who will give the valuable experience about their experience, their insight about the evaluation of the ATMPs product. And the first speaker is Dr. Monica Srivastava from TGA that will present TGA experience for ensuring quality aspect of ATMPs. And the second one is Dr. Jin Wukang from NIFDS, National Institute of Food Drug Safety Evaluation, that will present about the Korean experience on strategy for testing of ATMPs product. And after of each presentation, there will be a discussion. So in this session, the discussion will be conducted after each of the presenters or after each of the speakers. And now I would like to invite Dr. Monica Srivastava from TGA to present the topics ensuring the quality aspect of ATMPs. Before that, I would like to give a brief short information about Dr. Monica Srivastava. Dr. Monica Srivastava is a senior scientific evaluator in the biological section of India TGA. She already have more than 15 years experience, including PhD and postdoctoral position in molecular biology and immunology. So she possesses extensive technical knowledge in the field of molecular biology and genetic engineering, and enjoy using her scientific experience to ensure quality prescription medicines are available to the Australian publics. So to Dr. Monica Srivastava, the floor is yours. You can give the presentation, and your time is 30 minutes for your presentation.
Thank you so much for the introduction and the invitation to present in this workshop. I am just trying to find how to, share my screen. Just give me 1 second. Yeah,
So I will. Can people see my presentation now?
Yes, we can see your presentation, but maybe you can turn it into the slideshow.
Yeah, wonderful. So, I don't know why it changes into this one. Okay. Can you still see my slides in the slideshow? Is it better?
Yes, it's better. You can.
Wonderful.
It is the full screen now.
It is full screen. Can I start now, right?
Yes, you can start.
Oh, wonderful. Sorry. I will rely on your cues if there is any issues with my voice or with my slides, because I can't see any of you now in the audience. So once again, thank you so much for the invitation and allowing us to share TGA's regulatory approach for ensuring availability of high quality ATMPs in Australia. Myself, as I'm working as a senior scientific evaluator in the scientific evaluation branch of the Therapeutic Goods Administration, and the branch is responsible for pre-market evaluation of the prescription medicines, which ATMPs are also the prescription medicines, before they could be registered on the Australian Register for Therapeutic Goods and subsequently supplied in Australian market. The branch works very closely with other areas within the TGA, such as goods manufacturing practice, which is the GMP area responsible for licensing and clearance to the manufacturing site, the clinical evaluations that assesses the efficacy of the therapeutic products, and the post-market area, which monitors the products once they are supplied in the market. So ongoing monitoring of those products. I would also like to introduce Dr. Ekinola Edisa, who is also online with me, who will be joining me in the Q&A session. He's the principal evaluator also in the same branch. So how do we, a brief introduction about the Therapeutic Goods Administration. TGA was established in 1990, and all its regulatory powers are outlined in our TGA Act, established in 1989, and we call it as the Act. It provides a national system of controls relating to the quality and safety and efficacy of the therapeutic goods, and also their timely availability for use in and or exported from Australia. It's important to note here that the TGA regulates the therapeutic goods, so the products, and not the medical practice. There are other regulatory oversight for the medical practice, but TGA regulation is restricted to the final therapeutic product. Okay, so in my talk today, I hope to cover what Australia defines as ATMPs and how do we then regulate these cell and gene therapies, giving it a brief introduction about the different regulatory frameworks. And I will also discuss the manufacturing quality of the cell and gene therapy, specifically related to the evaluation of the technical data, highlighting some of the common issues and challenges that we see when we evaluate these therapies. So ATMPs in Australia, How do we define them? These are in Australia includes these are included as the gene therapies, gene modified cell therapies, and also cell and tissue therapies. So the definition that are used for these therapies is the substance that is used in or administered to human beings to either regulate, repair, replace, add or delete a genetic sequence, and the substance is involved in the therapeutic, prophylactic, or diagnostic effect of the product. So these are defined as gene therapies. Gene modified cell therapies, in other words, the cell based gene therapies, for example, CAR T cell, are also considered ATMPs in Australia. Other cell and tissue therapies that are not devices and have been classified as class 3 or class 4 biologicals, and I will go in a bit more detail about these classes later in my talk. And either A or B, any gene therapies or cell therapies, if they are provided in combination with a device, are also considered ATMPs in Australia. So ATMPs in Australia means all cell and tissue therapies and also the gene therapies. Okay, so regulation of advanced therapies in Australia, we don't have a dedicated advanced therapy network, sorry, the framework in Australia. We do have two separate regulatory framework to regulate advanced therapies. One is the biologicals framework, and the other one is the prescription medicine framework. Uh, Depending upon these frameworks are for biologicals or prescription medicine framework, depending on whether they involve ex vivo or in vivo manipulation of human cells. Both the regulatory framework have got different options for submission pathways. They have different GMP requirements, and there are different fee requirements and options, etc. So cell and tissue therapies are regulated under biological framework, whereas gene therapies are regulated under prescription medicine framework. Now let's have a closer look at the two frameworks. The biological framework. Under the biologicals framework, the biologicals are classified based on their risk profile. Class 1 being the low risk products, whereas class 4 are the high risk product. Anything that comprises, contains, or derived from human cells are considered biologicals. And this also includes things that comprises or contain live animal cells, tissues, or organs. For example, xenogenic transplantations are also considered biologicals. Some of the examples of the biologicals, the low risk class 1, 2 biologicals are fecal microbiota transplant products, skin products, demineralized bones, cardiovascular, etc., etc. And cell-based gene therapies, such as CAR T cell therapies, are regulated as class 4 biologicals, given the high risk associated with these products. And for my rest of my talk, I will be focusing CAR T cell therapy evaluation uh, further in discussion. So gene therapies fall under biological medicine, and they are regulated under prescription medicines framework. So the adeno-associated viruses or the lentiviral-based therapies are considered gene therapies, and the standards that are Australian specific requirements, as well as the ICH quality guidelines that are applicable to prescription medicine, is applicable to the gene therapies. They have different GMP requirements compared to cell and tissue therapies, and they have to comply with prescription medicine labeling order. Some of the approved gene therapies in Australia are Luxturna, Zolgensma, Hemgenix, Roctavian, um, and Roctavian. So the regulatory framework for both cell and gene therapy comprises of various international and national guidelines. And an example demonstrated here using the gene therapy, various ICH guidelines that TGA adopts and referred to during the evaluation phase. And the use of existing international guideline facilitates the convergence of consistent approach across the globe. It also reduces the regulatory burden by avoiding the reinventing of wheel related to the regulation of novel and emerging technologies. It's important to mention here as well that even though TGA has adopted various EMA and ICH guidelines, depending on the therapeutic, from time to time, we may consult non-adopted guidelines while evaluating cell and gene therapies. So the availability of these international guidelines does help to fasten the regulation of these therapies and eventually uh, uh, giving a timely availability of these therapies to the Australian public. Now let's have a look at the manufacturing quality of these cell and tissue therapy and gene therapies together. The manufacturing process of the gene therapy and the cell-based gene therapy have a significant overlap if you look at it. Gene therapy involves gene, vector, or a viral plasmid, which can be a virus or a plasmid, which is then manipulated, and after manipulation and undergoing various steps of manufacturing, it is injected in vivo into the patient. And one of the example is the Zolgensma gene therapy. Whereas the CAR T cell therapies are where the cells, mainly the T cells, are isolated from the patient and they are ex vivo manipulated either by using these gene therapy products. They are then expanded in vitro in the cell culture, and then once the ex vivo culture and modification and undergoing through a robust manufacturing process, they are transfused back to the patients. So considering this, you can see the distinct overlap between the two products, gene therapy and cell-based gene therapy. How we regulate them is slightly different, is the focus where the risk lies for the two products. For the gene therapy products, the risk is focused onto the end product, the final product, which is then tightly regulated by establishing robust release specification criteria. Whereas for the CAR T cell therapies, the risk is in the beginning for the donor selection and screening process. So that first step of establishing a good donor selection and screening process is critical for CAR T cell therapies. So one can see that the underlying evaluation of the cell and gene therapy has a lot of commonalities. The difference is in the focus of the risk between the two products. In the CAR T cell therapy, you do maintain the donor screening process, a robust donor screening process that constitute of having a medical history, a testing of infectious diseases, and the compatibility. Whereas for the rest of the three main critical attributes: the starting material, the in-process testing, and the final product is common to both the products. And I will be discussing them later in detail in my talk. Once again, what do we see during the evaluation of these therapies? So once the dossier is submitted to the TGA, the quality aspects related to both, as I said before, are very similar, other than the donor selection and screening process. You have the upstream manufacturing process comprising of cell procurement, purification, enrichment, and expansion and differentiation. And you do need to ensure that the donor selection is appropriate, purity of the product is established, and activation efficiency of the product is well maintained. At the same time, the downstream manufacturing processes, such as harvest and concentration, cryopreservation, quality control release testing, all the way up to the patient administration, is well controlled and defined. What is also important here to note is, apart from these technical details of the manufacturing process, you do have to ensure that the other information, such as the background information for the nomenclature, the naming of the product, the labeling of the product, is maintained. The questions that we ask when these therapies are submitted to the TGA is how well it is manufactured, how well it is characterized. Stability is an important part of the quality evaluation. Um, the stability testing is important and must document cells that are still viable and potent at the end of several hours required to release the product from the cell therapy. And this potency needs to be maintained during the transportation of this product, starting from the lab to the patient's bedside, and administer this product to the recipient. So you can see that in its entirety, not only the manufacturing process needs to be well controlled, but also the stability, comparability between the batch to batch, and also comparability between what was tested during the clinical trial phase all the way up to the commercial phase is critical. And at the same time, the consistency from batch to batch is also important. So from the quality aspects, all of these forms the critical attributes for assessment. Oops. All right, so the first important aspect from the evaluation of cell and gene therapy is the donor screening and testing. TGA requires the dossier to contain extensive details regarding the origin of the biological starting material and how it is handled, um, from the collection all the way up to the arrival at the manufacturing site. TGO 108 is a requirement. Compliance to TGO 108 must be demonstrated for allogeneic cell therapy products. Autologous cell therapy products are exempt from TGO 108. And TGO 108 have got a lot of details regarding the requirement, and that comprises of donor selection, medical and social history, uh, information about the mandatory infectious disease testing. Schedule 1 of TGO 108 also comprises of mandatory deferral periods, and also the documentation and ongoing donor management and review process needs to be well documented in the dossier. In certain cases, depending upon the situation, a gap analysis and adjustment may be acceptable through Section 14 um, part of the Act, which allows consent to vary from a particular standard. In addition to TJ 108 compliance, TJ 109, which is also order for biologicals, a therapeutic goods order for biologicals. Section 9 of the TJ 109 also describes the mandatory testings for the infectious disease safety and efficacy. Are there any donor conditions that may affect product quality and safety and efficacy should be considered and assessed. Next, we have the starting material. Um, Ensuring the starting material is of sufficient quality and consistency to manufacture a safe and effective product. Once again, the incoming batch of starting material must meet the material specifications. Transportation of a starting material again is very critical, and it must demonstrate compliance to TJ 109. And the defined limits of pre-processing, transportation, and storage, and these all storage and transportation conditions must be validated under worst case scenarios. For example, the temperatures, the time frame, they all must be validated real-time data under the worst case scenarios. Once again, ongoing management and review is also important for the starting material, and material variabilities and process variabilities needs to be documented. The importance of documentation of standard operating procedures, for example, any of the certificate of analysis documents that comes with the starting material, any of the process validation that is required with the starting material, any of the regulatory approvals for its use in clinical setting must be put in place. So altogether, the starting material needs to be well documented and validated, including the transportation and storage. Okay, so the third important attribute of cell and gene therapy is the in-process testing all the way up to the final product. Once again, if the viral vectors are important for both gene therapies and cell therapies, CAR T cell therapies, vector manufacturing process should form a discrete drug substance module in dossier. So it's a big part of manufacturing process altogether up to the stage of viral vector development. So that forms a part of the drug substance module. And per batch testing of replication competent virus, whether in vitro culture by using in vitro culture and passages, or by PCR based test are acceptable in the cell therapy stage. So very important testing, in process testing of the viral vectors. And then are the analytical procedures that are used. Selection of the assays that are used to demonstrate potency, purity, identity, all should be set and established after a thorough characterization studies of the vector. Once again, all the analytical procedures that are used to demonstrate these must be validated in compliance with the ICH Q2R2 guideline for analytical validation methods. During the process development, ensure all iterative steps in the manufacturing process are fully described and changes validated to demonstrate comparability of the product. Once again, emphasize the importance of comparability of the product here and the documentation of the from the process development, from the early stage of clinical trial all the way up to the commercial manufacturing. Changes to the manufacturing process of the vector, of course, once again, need to demonstrate comparability and of the final cellular product. The process, the product during the process development should be as close to the final product as possible. Many cell therapy products use a continuous manufacturing process, and sorry, I have a problem in here. And because of the continuous manufacturing process, there is often times when the drug substance and drug products cannot be differentiated because there are hardly any difference between the final drug substance and the final drug product. So the release testing is only conducted once, and of course the information can be presented either with the drug product module or with the drug substance module. So concluding this slide, the importance of in-process testing using appropriately validated analytical methods and keeping a robust record for the process development all the way to the final drug product is critical. Now, as much as the manufacturing process detail is important, labeling equally forms an important role in the quality, ensuring the quality aspect of the cell and gene therapy. Labeling requirements for biologicals are not the same as for biological medicines. Cell therapies such as CAR T cell follow the therapeutic goods order TG107, whereas biological medicines that have gene therapies follow TG91. So in this slide, taking an example of TG107, the information in this standard is about information that must be present either on the product label or any of the accompanying information. For example, instructions for use or product information for biologicals. It also covers, so the labeling doesn't only cover for the final product, the standard also cover information about the labeling of the biological starting material and donor to recipient traceability at all points in manufacture and supply. For advanced therapies, we also suggest the use of prescription medicine guidance for PIs, and this is common for both cell and gene therapy. And along with PI, the CMI, the consumer medicine information, is another helpful document and may be required from the risk management plan or the clinical evaluators. It's not mandatory, but except for class 4 biologicals where the CAR T cell therapy lies. So for CAR T cell therapy, risk management plan is mandatory. Yeah, so it is required for CAR T cell therapies to maintain a risk management plan. There is some information here on the approved cell and tissue naming and trade names. So the naming also forms an important part of the overall quality aspects. We do have an ACN application process in place. The proprietary ingredient for excipient buffers must be on the ingredient list. Trade names can also be included onto the ARTG entry, and they follow the ACN, ISBT trading nomenclature, which is you have your ingredient, active ingredient, followed by its processing, whether it's cryopreserved, whether it's fresh or frozen, following the storage, and its use, whether it's autologous or allogenic. And if it has a trade name, then the trade name will come at the end. So there are certain rules around the naming of these therapies, both cell and gene therapy, and there are certain standards that needs to be followed for complying with the labeling of the products. So, as I said before, evaluation of quality aspects of cell and gene therapy, we work very closely with other areas of the TGA, and there are different GMP pathways required available for gene therapy vectors and biological products. For example, gene therapy vector follow the GMP clearance pathway, while the biological product, which the CAR T cell therapy will be, require a GMP certificate for the manufacturing site. And there are differences in the process for obtaining these certification for the manufacturing site for these different product types. Manufacturing site for the gene therapy vectors are assessed under the PICS code for GMP. If these sites are overseas, it may be possible to obtain a clearance through a desktop audit using certification from another regulatory authority, provided appropriate agreements are in place between the regulatory agencies and the TGA, overseas regulatory agencies and the TGA. Once the human cells become involved in the manufacturing process, the manufacturing site will need to obtain a license for an Australian manufacturer or a TGA GMP certificate for the overseas manufacturer under the Australian Code of GMP. Again, this will require an on-site or remote inspection by the GMP team of the TGA. These certificates will be required for all the manufacturing sites, as we know that multiple manufacturing sites may be involved for the development of cell and gene therapies. So the certificates are required for all manufacturing sites, including those that conduct the donor testing or the release testing of the final biological products. There are links provided in this table that provides more detailed information about the GMP requirements, but nonetheless, it is suffice to say that along with the quality aspects of the cell and gene therapy, GMP plays an important role in ensuring high quality ATMPs in Australia. So now we have seen certain aspects of regulating TGA ATMPs in Australia. And I would like to now highlight a little bit of, because as I mentioned before, we are in the scientific evaluation branch of the TGA that looks after the pre-market evaluation of these therapies. I would like to point out the importance of post-market monitoring and RMPs for these products as well, even though our branch is not directly involved in those things. TGA definitely as a whole regulates the post-marketing monitoring and RMPs for these therapies as well. So the risk management plan is important for ATMPs involving well-maintained patient registries and mandatory reporting for adverse events. And class 3 and class 4 require a risk management. So as I said, it's mandatory to ensure ongoing safety and efficacy of these products. And the risk management plan includes safety specification, product vigilance plan, and a risk minimization plan all together that helps TGA put in a biovigilance monitoring for the product once it is in circulation in the market. So yes, again, the post-market monitoring and RMP processes such as RMP help ensuring the quality of ATMPs. Now, I would like to now discuss some of the technical issues and some of the common issues that we come across during the evaluation of cell and gene therapy. So, bringing first the gene therapy, once again, there's few key attributes that needs to be looked at. Is it well characterized? Which means that the the requirements of having an elucidation of the structure. Is the amino acid sequence determining the proteins is correct? Is the in vitro infectivity is happening? Is the nucleotide sequence is correct? Again, the second most important is the purification. Are the impurities such as cell component, media, and transcription reagents removed? Encapsulation of non-target sequences, elimination of these sequences is important for a pure preparation of the gene therapy. And number 3 is the potency assay. The choice
Dr. Monica, are you still joined?
Sorry, Monica, I think you have to unmute.
Okay.
Monica, you are muted.
Dr. Monica, we cannot hear your voice.
Mute. Okay, can you guys hear me?
Yes, we can hear you now.
And can you see my slides? I don't know what happened there. Apologies for that.
Okay, we can see your slide, but could you please do the slideshow?
Okay, I hope it works. Can you see the slides?
Yes, we can see, but could you please
Yeah.
do the slideshow? Okay.
Yeah. I have no idea what happened there. Apologies. And is it, how far do you want me to go back?
About the challenges for the gene therapies.
Okay, so we are here. All right. All right. So as I was saying before that during the evaluation of gene therapy, a couple of the critical attributes that we focus during the evaluation or the assessment of the technical information is the elucidation of the structure, the purification of the product, and finally the potency assays that are chosen for various viral vector. In terms of evaluation of CAR T cell products, we do have, again, overlapping, assessment of the stages. For example, control of starting material, which is donor apheresis. This is limited to the evaluation of CAR T cell product. However, the second paragraph on the viral vectors for CAR T cell product is again common for both cell and gene therapy. So control of starting material in terms of donor apheresis, and for autologous product, TG 108 does not apply. But having said that, it doesn't mean that the autologous product does not have risk of contamination and the infectious disease. So it becomes very critical to assess autologous products um, um, and assess the risk of contamination between batches if infectious disease status of the donor is unknown. In those cases, we rely on our GMP partners, ensuring that the manufacturing sites have got appropriate processes and procedures in place for traceability, for documentation. Once again, the cryopreservation procedures, um, the validation of the manufacturing, such as fresh versus cryopreserved starting material, and of course, the transportation from the collection to the manufacturing site, all needs to be very well validated. Um, Once again, where the TGO 108 compliance cannot be considered, we, we try to consider TGO 109, which is again a biological standard. And there are again, are there any diseases or conditions that may affect the quality, safety, and efficacy of the final product? Is the question we ask. And is the manufacturer require minimum material specifications, for example, are put in place? For example, the viability of the cells, number of CD3 positive cells, that is the starting material, and appropriate testing of the starting material, such as bioburden test. So these are the important controls that we evaluate for the starting material for CAR T cell therapies. In terms of viral vectors, um, whether it's lentiviral or retroviral vectors, evaluating using the guidelines for gene therapy vectors. The vectors, each batch of the vector must be tested for RCL using the in vitro culture method, and potency measures should be in place using validated potency assays, and the purity of these vectors. Some level of control and specification must be put in place around the particle to PFU ratio in plaque assays. Now, one of the key issues for us in evaluation of CAR T cell product is also the product release testings and the specifications set around the final product. We can acknowledge that the autologous products will be variable in quality because governed by the highly variable nature of the original starting materials. So the specifications around these autologous products are usually wide, and this is acknowledged. There is a risk that batches not meeting release specification will result in patient death if they cannot get the product. So we need to come the risk versus benefit analysis in those type of scenarios. Whatever be the case, the specifications that are set must be justified and based on the manufacturing experience. So one of this thing with the autologous CAR T products is around the product release testing and specifications, the wide specifications that we observe. Basic safety specification standards should always be there. For example, sterility and endotoxins are part of the release testing and specification. Per batch testing of replication competent vector is also important. If there is sufficient manufacturing history, then PCR-based methods are acceptable. Vector copy number per cell is less than 5 vector copy number per cell is acceptable, and dose is calculated based on the total number of viable. So the viability of the CAR T cells is important, and this should be specified and justified based on the purity levels of the CAR T cells. The
Uh,
viability.
sorry,
Hello?
Dr. Malika, your time is 5 minutes again.
Sure. Okay. Um, uh, I'm almost there. So there's, I think, 1 or 2 slides left. So once again, CAR T product release testing and specifications, we identify that for the autologous products, we do have certain challenges to overcome, but nonetheless, there are set guidelines and standards for uh, the release specifications around specific standards such as sterility, purity, viability, endotoxin, etc. Um, Further to those, again, emphasizing that the testing of residual impurities for vector drug substance is important, and residual non-T cell tested using flow cytometry should be well justified and should be within the limits. Again, I will not go in detail the potency. The potency must be linked to the product characterization, and there should be additional assays put in place, such as cytotoxicity and cytokine release, interferon gamma assays, to comprehend the overall potency of the product. All release tests must be validated according to ICH guidelines, Q2R2. And some of the relevant quality evaluations of the cell therapies. So that these are the standards and the AMA guidelines that I have provided here. And some of the quality evaluations guidelines, ICH guidelines for the evaluation of gene therapies are provided here for your assistance. And that's it. That's the end of my talk. And thank you for listening.
Yes, thank you for your presentation, your completed presentation. And now we are going to the next session, which is the discussion. And here with us, also your colleague from Australia, Dr. Akinola Adisa, join in this Zoom meeting. Welcome, Dr. Akinola. And
Thank you.
okay, good morning, Dr. Akinola, and we can start the discussion. Is there any question from the audience? Please raise your your hands and say your names and what question do you want to ask? Okay.
Yes, thank you for your fruitful presentation. I'm Dia from Biological Product section. I'm also the evaluator. First, I would like to ask about labeling, special labeling for biologicals on ATMPs that TGA said that you already put the information on the uh, for starting material source and for donor to recipient traceability. And then I would like to ask whether is the batch number on the packaging traceability, or and what kind of the example of the labeling info for this traceability that TGA apa TGA asked to the pharmaceutical industry to provide this information. That the first question. And then for the second one, I would like to ask about secretum. I think secretum is not included in the ATMPs, but since I look at your CV that you are a biological product evaluator, I think it's a general. I would like to ask, what is the quality aspect of the secretum? I think you already know that secretum is the molecule or biological factor that's secreted by cells into the extracellular uh, environment. And then I would like to ask, is the quality aspect of the secretum can be referred to the ATMP's requirement for the quality, or is there any specific requirement to treat the secretum for the quality aspect evaluation. And then the third question, if you don't mind, I would like to ask about the CAR T cell that you already presented, that the viability number generally for the CAR T cell is generally more than and equal to 70%. And the question is, I think it's quite a wide range. And I would like to ask whether if the production batch for this viability is in the wide range, the percentage for viability, is it needed to adjust the doses that give to the patient according this viability for the CAR T cell? Thank you. I hope my question is clear to you.
Yes, to Dr. Monica, you may answer the question for Mbak Dia. The first one is about the labeling. The second one is about the quality aspect of the secretome that to be submitted in the registration process. And the third one about the CAR T cells, about the it is needed to adjust the doses when the viability is quite range between 70 until 100%.
Thank you for the question. Look, if I understand the question correctly for the labeling, so I can't understand the question correctly for the labeling. What do you want to know about the labeling of the biologicals? We do have TGO 107 compliance to demonstrate it for the labeling of the biologicals. That includes CAR T cell therapies, and which means that there are some mandatory requirements for TGO 107 that needs to go on the label. And the label in this sense, the label is also the label that goes onto the starting material, which is the donor collected cells, and also to the final product. So
for a final product.
For the final product,
Yes,
and it's for both.
yes,
And it is very important to have these labeling put in place. That includes the batch number, that includes the product name and description. And it is very important to have this from the starting material onto the final product to maintain the traceability of the product from the product back to the donors. Did that address your answer? So the
yes. I thought that in the final product there is additional information on regard for the donor to recipient traceability. My question is for product. I think every product has the specific batch number. I would like to ask, is the batch number on the final product is not enough to trace the about this traceability, or we need any additional information on regard to donor in the final product labeling?
So there are certain things that are mandatory, for example, the batch numbers, but there are things that can assist the sponsors for traceability, and if there is a space on the label, that can be included onto the batches, onto the label, but they are not mandatory. So anything that assists.
Can I add a little bit to that? This is Sakin Adisa speaking, also from TGA. Can you hear me?
Yes, we can hear you. We can hear you.
Right. Thank you. So as Monica explained during the presentation, two different, I mean, not essentially two different, but separate framework for ATMPs generally. So we have the prescription medicine option, which is when you are going down the path of gene therapy without involvement of cells, or if you have cells involved, such as CAR T cell, then you go down the route of biological framework. So each of them have got labeling requirements depending on what you are looking at. So if you just take the biologicals, the CAR T cell example, for instance, we are dealing with autologous product. So it's taking, sorry, autologous product. So it's taken from one patient, manipulated, and then back to the same patient. So traceability in that case is fairly straightforward because this product will never go to any other patient. So the requirement for the label will probably be fairly standard, straightforward. You might have batch number, maybe to record information around how it was made and then back to the same patient. However, if you must go down the route of halogenic, that is, you take a cell from a patient, you manipulate it, and it can end up in other patients, then you could expect that you will need to have additional information on the label so that you can tell that this product starts from this patient but ended up in many other patients. So batch number, we have matrix code, serialization code that if sponsors choose to, they can put on that label. But if they choose to put that 2D barcode on the label, then we have standard to guide that as well. Okay, so that is on the cell therapy side. But if it is gene therapy, we have even a bigger requirement when it comes to label because that falls under the prescription medicines arm. And in that case, so you again batch number. If you have 2D matrix code, you need to follow a certain standard, but then you need to present information in a certain way. So altogether, yes, we do we put a lot of attention into traceability to make sure that, I mean, we put the onus on the industry anyway to make sure that when something comes out of the manufacturing plant, they need to be able to trace it from the source to the patient. And there are bits and pieces, not just the labeling, but other aspects of the regulatory process that ensures that that happens. So I
Yeah.
wonder whether that answers your question.
Is it clear? Yes, I think for the first question is quite clear about the labelings, and we can move to the second question about the secretomes. Maybe Dr. Monica or you, Akinola, can give us the answer.
Yeah.
Monica can answer that. Thanks.
Yeah. My apologies again. I have not heard the secretomes coming to the TGA yet, so I'm not sure this product. We have seen this product yet, but if it comes, as I said, if it meets the criteria defined for the biologicals, if it contains or derived from human cell and tissue, it is very likely to be regulated as biologicals at the TGA. Are these secretomes you are referring to as the exosomes? Are they equivalent terms or is it different?
Yeah, it quite similar because in Indonesia there are many developments of this product secretome. So I would like to ask whether the ATMPs requirement on quality can be applied to this product.
So look, based on very little information about the product itself, and as I said before, that we have not seen this coming to us, very likely it's going to be biological and it will be regulated under the biological framework, depending upon how it is manufactured and how it is used. So allogenic or autologous, it may fall into either of the classifications of for biologicals from 1 to 4. Once again, depending upon the product, how it's manufactured and what are its use, it is very likely to fall in one of those classifications for biologicals and very likely to be regulated as biologicals.
Okay, Monica, is it clear, Badia?
Yes.
Because there are no experience yet about the secretum, right? But It depends on the manufacturing process, which is who determine which is the guideline would be for the evaluation of the quality. Is it correct? Okay.
True.
Now we
Great.
can move the third question about the CAR T cells, that is it needed to adjust the dosage since the viability of the CAR T cells is quite range. The range is quite large, from 70 until 100%. Is it we need to adjust the dosage?
Look, again, depending upon the various factors, again, yes, the dosage should be adjusted based upon the viability of the final product, depending where in the wide range of viability it lies. But I would also say that there is a lot of progress happening in terms of getting a more viable product and getting the viability towards from 70% to higher levels. And that is another thing that needs to be adjusted in terms of the manufacturing process of these products as well. So yes, dosage is one component that needs to be adjusted depending upon the viability, but also the overall manufacturing process development should also be adjusted to aim for higher viability of these products so that the dosage remains the same.
Okay, Dr. Monica, Dr. Akinola, I think the third question is quite clear. It is depend on the manufacturing process if we needed to adjust or not about the dosage. Okay.
Yeah.
Now because the time is runs and we are almost to the end of the session for TGA, and I would like to give the MC for the closing.
Okay, thank you, Dr. Srivastava, for your excellent presentation. Thank you, Dr. Adisa. But before we ending this session, in recognition of Dr. Srivastava's outstanding contribution, we would like to present her with a certificate of appreciation.
Thank you very much.
This certificate symbolizes our gratitude and acknowledges her contribution. And please give a round of
Okay, Dr. Monica, Dr. Akinola, thank you very much for your very comprehensive information related to ATMPs in this session. We now can move to the second session with Dr. Jin Wukang. Welcome. Good morning, doctor. And for you, Monica, Akinola, if you want still join with us, please join. But if you still have another schedule, you can run your schedules. Okay, thank you.
Thank you so much for the invitation, and I will take my leave now. Bye bye. Have a wonderful
Bye.
time.
Thank you.
Thank you. See you.
Thank you. See you.
Bye.
Okay, for the next speaker, Dr. Jin Wungkak. Welcome, Dr. Jin Wukang. She is the evaluator at the NIFDS Korea. She will be presented the topic strategy for testing of advanced therapy medicinal product. First of all, I would like to give a brief short notice information about Dr. Jin Wukang. She is the senior scientific officer in the MFDS, and she works from 2004 until now. So she has a lot of experience, almost 20 years in the fields of biologicals. And she already have several publications related the stem cell. And now she will give her experience about the evaluation of the stem cell in the MFDS Korea. So, Dr. Jin Wukang, please, you can start your presentation. The floor is yours.
Hi, good morning. Can you hear me?
Yes, I can. We can hear you.
Okay, I will start. Yeah, I'd like to thank the organizer for giving me the opportunity to present today. By way of an introduction, I am Deputy Director of Cell and Gene Therapy Product Division from NIFDS in Korea. Next slide. Next slide, please. Thank you. Please understand that the following represent my views based on my experience and not necessarily the view of Korea MFDS. Next slide, please. These are three topics I will talk about today. Next. First, I will talk about the regulatory basis and characteristics of advanced biological products, which are also called ATMP. Next slide. This slide is our regulatory framework related with advanced biological product. In addition to the Pharmaceutical Effect Act related with general drug, the Korean new law Act on the Safety of and Support for Advanced Regenerative Medicine and Advanced Biopharmaceuticals was legislated and enforced in August 2020. Because of a huge need for regulation of a new concept, which is produced in small quantities and mostly target rare disease and need special safety control. This new act takes preference over General Pharmaceutical Affairs Act. Below the law, there are regulation, the MFDS notification, and guideline. Next slide, please. These are types of advanced biological product in Korea. Main products are cell therapy product and gene therapy product. My talk today is focusing on testing strategy of cell therapy product. Next slide. The structure of cell and gene therapy product is very complex compared with the small molecule and protein like growth hormone and monoclonal antibody. Maybe this is like the Battle of Star Wars. As the structure become more complex, the more regulatory considerations become necessary. Next slide. Advanced biopharmaceuticals are large molecular size and structural complex compared with the synthetic drug and produced by complicated manufacture using living cells or viruses. We should pay extra attention to the controls of contamination and impurities because it is greatly affected by environment and sensitive to changes. Next slide. Cell and gene therapy products are produced by the manufacturer with the human cells and raw materials. Next slide. In addition to the initial donor or patient of cell and tissue, these ancillary raw materials also originated from animal or human. It should be manufactured by the materials which do not contain any TSC, BSC contamination and any kinds of viruses. Oh, last slide. Last slide. Before? Yeah. Thank you. This risk from animal or human-derived materials could be challenging to advanced biological products. Next slide. The last one. Oh. Forward. Another challenge of advanced biological product is the manufacturing process. When you produce cell therapy or gene therapy product, it is not possible to do either viral clearance or stereo filtration. Automated filling is also highly challenged. Next slide. Next, I will talk about the guidelines on test and method when we release the cell therapy product. I want to present the guideline on release test for cell therapy product. I will talk about all the test specific consideration in detail. Next slide. The scope of this guideline is about the data requirement for quality assessment when you do release test for cell therapy and ex vivo gene therapy. Next slide. Unique feature of cell therapy product should be considered in selecting release test and method to ensure the safety and efficacy of the product. Release tests should be conducted at the drug substance or drug product stage. Generally, drug substance should be established at the stage where the preparation of cell culture containing cells is completed. Next slide. In the appearance section, the basic outward appearance of a product should be examined. For injectable, the primary container should also be written, and the color and physical state of the drug before and after freezing or reconstitution should be observed. Next slide. Sterility test result should be interpreted in association with the result from environmental monitoring and IPC test. In principle, sterility testing should be conducted on each lot of drug substance or final drug product before initiating the filling process. Next slide. Cell culture-based rapid sterility tests, which are non-compendial methods, are also permitted for use in sterility testing. We can determine microbial growth in microbial cultures using superior to the medium that is used for compendial method. In case of using rapid tests, the validation of analytical procedures and comparability to compendial method. For suitability testing for sterility, the same type of metrics as the same metrics should be used for sterility testing. Because slower growing strains and frequently detected by environment monitoring in the manufacturing site, all strains, including the compendial strains, should be confirmed under the same test conditions for minimum 7 days. Next slide. Because autologous or one-to-one donor-derived cell therapy products were manufactured and filled as one-time treatment, so we cannot help performing sterility release test with a limited volume. So through our research project, we established the minimum test sample volume based on the fill volume of the final product. Next slide. In the mycoplasma testing, should be conducted MFDS notification or compendial method. Validation, such as growth inhibition activity assay and media performance testing, should be conducted beforehand. Mycoplasma testing samples are used with both the final cell culture media and cells prior to washing. We could use nucleic acid amplification technique, which is NET test, in case of confirming to be equivalent to compendial method. The mycoplasma testing result may show false negative because they have been lost during DNA extraction. So in preparation for such cases, each test should include extraction positive control. Next slide. Endotoxin test spec should be established considering the administration method and duration and body weight. In case of test method, Korean compendial method should be used, including these three methods: gel clot, turbidimetric, and colorimetric method. And preliminary testing for the respective three methods is required to ensure the specificity and effectiveness. Endotoxin testing should be conducted per each lot using validated test endotoxin. If the test results are not deemed trustworthy, the club method should be used for the final conclusion. Next slide, please. Contamination may arise mostly from the raw material during manufacturing or operator. Adventitious virus testing is one of the most important tests. It should be used with both the final cell culture media and about 10 million cells prior to washing. The concrete adventitious in vitro test method is using 3 types of indicator cell line, culture for 14 days and consecutive culture for another 14 days, total 28 days, 3 types of red blood cells used. We can detect the adventitious virus by cytopathic effect, hemagglutination, hemabsorption reaction. In case of using the animal origin material with high risk of viruses, species-specific virus detection testing is required. In case of cell bank or drug substance exceeding a usual size, in vivo adventitious virus test is additionally required. Next slide. In case of cell therapy product for one individual, such as autologous or one-to-one donor-derived cell therapy that satisfy the conditions below, we can exempt the adventitious virus testing. Animal or human-derived materials with compendial safety standards were verified by viral testing appropriate or viral clearance process. Adventitious viral test result on final product from 3 lots produced in GMP manufacturing site within the acceptable range. Next slide. For in case of allogenic cell therapy product, not autologous cell therapy product, that satisfy the conditions below, we can conduct adventitious viral testing periodically. Viral safety testing should be conducted appropriately at the cell bank stage. And materials are at very low risk of viral contamination, and test results are within the acceptable range. Even when adventitious viral testing is not included in the quality testing package, if change occur in the manufacturing method, the change should be justified by 3 consecutive lateral results. Next slide. Okay. Because cell therapy products are manufactured by small scale or all the cells prepared and administered to the patient, it is difficult withholding release testing. In this case, using a portion of the final preparation live before filling may be considered. For cryopreserved cell therapy product, samples for release testing should do freezing, thawing before the testing. And for in case of organoid product, in addition to the number of cells, the number of aggregate should be counted. Next slide. The objective of identify is to confirm the identity of the cells to be intended as an API, active pharmaceutical ingredient. The basic properties of the cells should be identified using various markers and assays such as morphological, immunological, and biological assay, including appropriate positive control or negative control. If additional elements such as a scaffold or extracellular matrix should be also identified. Next slide. The objective of purity is to identify the absence of residual undesired substance in the final product. We should investigate the use of materials that may pose safety concerns during manufacturing process and after the completion of manufacturing. We should determine the need for further control to ensure that their remnant will remain below appropriate levels. For example, stem cell therapy product, undifferentiated cells or differentiated into non-target cells or feeder cells should be controlled as purity testing. Next slide. The objective of the potency is confirming the proposed efficacy of a drug product. Potency markers should identify biological activity of the product and should be evaluated by quantitative method. Selected markers should confirm the clinical therapeutic effect of the cells on specific disease and based on the mode of action. From the planning stage of the clinical study, it is important to monitor the relevance of potency testing and clinical outcome. And rather than one method of potency test, it is necessary to establish several different markers to allow for a comprehensive assessment of the therapeutic mechanism. Next slide. In case of general testing may be waived if scientifically justified. For example, cell size over 10 micrometer, it is meaningless to do foreign insoluble matter test or insoluble particulate matter test for injectable product. In this case, general test for excipient will be required instead of final product. Next slide. As we move on to the last topic, the reference guideline list. Those guidelines is our regulator and guideline about quality review of cell and gene therapy products. Guidelines can be found on the Ministry of Food and Drug Safety website. And next slide. Those are WHO and ICH quality related guidelines. And next slide. Those are FDA. Next slide. And FDA and USP quality related guideline with reference. Thank you for your attention.
Okay, thank you, Dr. Jin Wongkang for your presentation. And now we can continue to the session for the discussion. Is there any question from the audience related the testing, strategy testing of the ATMPs products? Is there any question? Is there any question? Because this is quite important because it's related to the quality aspect of the ATMPs that we should be evaluated in the registration process or in the production process. Fine. Okay. Yeah, okay, finally, yes. You can say your name and the question you want to ask. Okay.
Yes.
Thank you for the great presentation from Dr. Jin Wongkang. My name is Angliana. I'm from Prodia Stem Cells. I would like to ask about the adventitious virus testing that you are recommending. Is there any specific adventitious virus that need to be tested for the cell therapy product for specifically? Thank you.
We don't know the specific virus of adventitious virus test. So we should the protocol. As I mentioned before, we should do 3 types of indicator cell line. This is the type of cell type which is the same as the cell therapy product, the same type, and human fibroblast cell and monkey cell. And we should culture 14 days, and after these 14 days, we can culture consecutive culture for another 14 days, because the remnant virus can hide in the cell therapy product. So we should culture 28 days total. Yeah. Can you answer the question? Can I answer?
Yes. Is it clear for the answer? Is there any other one more question if you want to ask to Dr. Jin Wongka? I think it's great opportunity to ask the question to the real to the person who experienced about the gene therapy. Is there any question? Yes.
Hello, good afternoon. My name is Henry. I'm from Dewung Biologics Indonesia. Thank you for your comprehensive and also insightful presentation. I have a question related to the validation. So in your presentation, you mentioned that for the mycoplasma, it is possible for us to use the validated nucleic amplification or NAT to replace the culture-based method. And do you have any best practice in order to perform that, whether we need to perform the regular one and then compare it the using compendial method, or is there any best practice for that? Thank you.
Okay. Dr. Jimukan, can you have the clear information about the question, or we need to repeat again? I think the question is, do you have a best practice or the guideline for the to determine which is the to determine the specification of the gene therapy or cell therapy? Is it correct, Dr. Henry, or you can repeat your question again?
Okay, we have a guideline of net test for the cell and gene therapy product. So you want to the guideline? I can send you. Because but this is the Korean. We should translate. Yeah.
Unfortunately, the guideline is still on Korean. Yes, I think our handphone already have advanced technology to to translate automatically. Is it clear, Pak Henry? Okay. Is it the time we can? I have signs from the MC. So thank you, Dr. Jim Kang, for your excellent presentation today and your sharing experience in the cell therapy. And now I deliver the next session to the MC.
Okay, thank you so much, Dr. Kang, for your insightful presentation today. In appreciation of Dr. Kang's remarkable work, we are proud to present this certificate of appreciation. Let's give a warm round of applause.
Okay, thank you, Dr. Jin Wukang. I think this is our last session in this afternoon, and because it's in our time is is the time for the lunch. I think we can end this session. Thank you very much for all the speaker and all of the audience. Uh, I hope this opportunity will give us some good or new information, and with it will be improve our knowledge about the ATMPs. Thank you very much for all audience. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Terima kasih kami ucapkan untuk Ibu moderator yang telah memandu sesi pada siang hari ini.
Boleh diberikan tepuk tangan dulu Bapak Ibu?
Tentu juga kami ucapkan terima kasih atas antusiasme peserta workshop untuk berdiskusi dengan narasumber kita pada hari ini. Boleh kita beri tepuk tangan sekali lagi untuk kita semua. Hadirin yang kami hormati, jam sudah menunjukkan waktu makan siang. Kami persilakan Bapak Ibu untuk menikmati hidangan makan siang yang telah tersedia pada ruangan restoran di sisi kiri lift. Bagi Bapak Ibu yang akan menunaikan ibadah sholat zuhur, dapat menunaikan sholat di musola lantai 10.
Bapak Ibu peserta yang hadir secara luring dan daring, acara akan dilanjutkan pada pukul 13.30. Kami harapkan Bapak Ibu untuk dapat kembali hadir tempat waktu. Terima kasih kami ucapkan selamat menikmati makan siang.
atau nggak ada dari manusia gitu kan. Oh, nggak ada foto itu. Ini segede apa? Gini.
Tes. Tes 1, 2, 3. Bapak Ibu peserta workshop yang kami hormati, acara workshop akan dimulai sebentar lagi. Jadi kami mohon Bapak Ibu untuk segera memasuki ruangan acara. Sekali lagi, Bapak Ibu peserta workshop yang kami hormati, acara workshop akan kita mulai sebentar lagi. Jadi kami mohon Bapak Ibu untuk segera memasuki ruangan acara. kepada rekan Ane, saya tunggu di depan di meja asrot. Kepada rekan Ane, ditunggu di depan meja asrot. Oke, baik. Selamat siang Bapak Ibu peserta workshop yang kami hormati. Masih semangat siang ini Bapak Ibu? Saya juga menyambut teman-teman yang hadir di daring. Acara workshop akan segera kita mulai. Baik Bapak Ibu yang terhormat, sebelum kita mulai acara workshop di sesi selanjutnya, di sini ada teman saya, Ibu Ane. Ibu Ane akan memandu sesi ice breaking dulu sebelum kita mulai lanjut sesi workshop selanjutnya. Boleh kita tepuk tangan dulu dong Ibu?
Sebelumnya kepada Bapak Fuad minta tolong untuk share screen.
Kita mau ngapain nih Bu Ane?
Kita akan kuis nih siang-siang sebelum melaksanakan workshop selanjutnya. Kita bikin otak kita fokus dulu. Kita kuis dulu, tapi kuisnya nggak susah kok. Bukan tentang ATMP, tenang aja. Kuisnya gampang, insya Allah. Dan berhadiah menarik ya.
Ada hadiahnya?
Ada dong. Untuk juara 1, 2, 3 dapat saldo OVO. Jadi ayo berlomba-lomba untuk menjadi pemenang supaya mendapatkan hadiah.
Iya, yang dibutuhkan adalah handphonenya,
Handphonenya, ya betul. Bisa ketik di Google kahoot.it, kemudian memasukkan pin game yang ada di atas ini 294 423, terus masukin namanya.
Kita juga mengundang teman-teman peserta workshop yang yang mengikuti secara daring, bisa ikut juga kan ya?
Boleh, bisa banget, bisa ikut.
Ada QR code-nya juga bisa di-scan mungkin ya?
Ya, bisa juga di-scan melalui QR code yang ada di layar, tapi kalau misalnya teman-teman yang di ruangan terlalu jauh, bisa pakai pin game 294 423.
Nampak di layar peserta yang mengikuti secara daring ada 82 dan secara luring ada 2.500 sepertinya ya. Jadi kita tunggu sampai berapa ini? Sudah 40 peserta yang sudah ikut. Oke, namanya ini ya, beraneka ya. Saya nggak kenal sepertinya ini namanya. Oh iya.
Wah, ada STY.
Iya. Dari teman-teman perwakilan universitas juga bisa bergabung yang mengikuti secara daring.
Udah siapa lagi? Udah siapa lagi nih ya?
Oke, kita tunggu sampai 100 bagaimana?
Oh, boleh. Ditunggu sampai 100 ya. Setelah itu kita coba mulai. Jadi ayo segera join secepatnya karena hadiahnya sungguh menarik ya, lumayan. Buat jajan-jajan yang baik ya.
Oke, sudah masuk ke angka 70. Masih bertambah?
Ayo, apakah sudah join semua teman-teman yang di ruangan?
Ada Fufu Fafa kok saya.
Waduh, ada Fufu Fafa. Ada Jungkook loh Mbak Eca.
Aduh, Masya Allah. Kita ini beraneka sekali pesertanya hari ini.
Iya, dari berbagai macam
golongan ya.
Golongan, betul. Ayo, sudah 82.
Makin melambat di 82, 83.
Siapa lagi yang belum join? Apakah sudah join semua?
Sepertinya kalau makin melambat kita tahan di 90. Oh, tapi masih bertambah.
Oh,
ya?
sekarang ada Fafa Fufu, temannya Fufu Fafa.
Ada.
Baik. Kembarannya mungkin ya.
Oke. Nah, udah di 87.
Sudah semua join? Kalau sudah, mungkin kita mulai
Oke,
saja.
kita mulai langsung aja ya,
Ya.
Mbak Diane ya.
Oke, Mas Fuad, mainkan. Suaranya Mas Fuad? Nah, yang pertama nih
Tiwi
pertanyaannya.
sedang menaiki KRL menuju Depok. Bila KRL tersebut melaju ke arah selatan, kemana asapnya katanya?
Ayo, kemana arah asapnya?
Ayo. Mungkin suaranya bisa di-share, Mas. Ya. Ayo, kemana katanya asapnya?
Kemana nih?
Ini
Ke utara kah? Ke barat daya?
Soal CPNS banget.
Share lagi. Share lagi.
Ya, gimana ini gimana?
Wah, Mas Fuad gimana nih?
Wah. Ini.
Oh,
Udah 49 orang
ada
jawab benar.
satu yang salah ini.
Enggak, 38 juga salah Mbak
Oh,
Eca.
38 ya, 38.
Banyak ya yang
Banyak
salah.
yang salah ya.
Oke, coba kita lihat perolehan siapa yang tertinggi.
Woi,
Anita Putri.
Ini paling cepat berarti.
Cepat jawab. Kita berikutnya Mas Fuad.
Tebak nama artis.
Nah, siapakah? Kira-kira siapa nih nama artis?
Ada 2 gambar ini.
Ada lemon.
Yuk, ketikkan jawaban Anda katanya. Ada lemon.
Ada orang.
Ada emoticon.
Ada emoticon.
Emoticon seperti ini.
Kira-kira siapa nih nama artisnya?
Ini memutar otak ya.
Siapa ya?
Apa ya?
Siapa ya? Limbat, Bapak Ibu.
Oh, limbat, limbat gitu ya.
Betul, Mbak Eca, limbat. Jadi
Limbat.
13 orang benar loh.
13
luar biasa.
Out of the box, hebat ya.
Hebat.
Tepuk
Kita
tangan
lihat
dong buat 13 orang.
Kita lihat siapa yang memimpin.
Oh,
Sius.
Katanya Sius.
Oke, selanjutnya Mas Fuad.
Oh,
ini
iya,
ada
itu limbat.
artisnya ya.
Ini limbat tuh yang ini ya.
Iya, betul.
Apa kepanjangan TB pada nama TB Simatupang? Nah, Tribonar, Tuanku Bagus. Jawaban yang lain. Kira-kira yang mana? Kepanjangan dari TB dari nama TB Simatupang.
Wah, 31 orang benar. Terkocek dengan tuh bagus ya.
Oh iya.
Yang benar adalah Tribonar.
Iya.
Siapakah yang memimpin? Oh, masih Sius. Luar biasa Sius.
Sius hebat ya.
Hebat.
Oke, ayo pertahankan Sius. Oke, pertanyaan selanjutnya. Jika gajah jadi ayam, harimau jadi ayam, ular jadi ayam, lalu ayam jadi apa? Katanya gitu.
Ayo, jadi apa ayamnya ya? Kalau semuanya jadi ayam jadi apa?
Pertanyaannya bapak-bapak banget ya. Jawabannya adalah jadi banyak ya.
Iya betul, tapi masih ada yang salah ternyata. Tidak apa-apa. Tidak apa-apa. Berikutnya.
Masih ada yang jawab.
Siapakah yang masih? Oh, Sius masih memimpin. Luar biasa. Sius.
Saya penasaran ini sama Sius ini.
Hewan apakah ini? Nah, kira-kira dari kakinya nih hewan apa ya?
Oh, kaki ya itu. Oh, hewan apakah ini? Buaya, badak, kucing, atau katak?
Ayo, hewan apa ya kira-kira?
Katak bukan ternyata.
Yang benar buaya.
Katanya.
Menjawab benar, siapa yang memimpin? Masih Sius. Coba Mas Fuad bagaimana gambarnya? Nah, iya teman-teman.
Ini masuk ke gene modification ya, sepertinya ya. Iya, terserah aja deh pokoknya ya.
Kita lanjut ya soal berikutnya.
Jika seekor kuda menghadap selatan, maka ekornya menghadap, tenggara,
ekornya?
atas, bawah, atau utara.
Kalau kudanya ke selatan nih, ekornya kemana nih ya?
Ini mikir banget sepertinya demi saldo OVO ya.
Wah, luar biasa. Mayoritas benar nih Mbak Eca.
Menghadap ke bawah.
Betul
Bagus.
sekali.
Hebat.
Tidak terkecoh, bagus.
Tidak terkecoh.
Wah, masih
Sius.
Sius yang memimpin. Keren nih Sius.
Oke.
Lanjut Mas Fuad.
Ada 4 soal lagi.
Tebak nama pelawak nih sekarang. Nah, ada 2 gambar lagi.
Saya senang nih kalau kayak gini.
Ini
Apa
ada
ya?
emoticon tangan ke bawah. Sama ada gambar kucing. Kira-kira siapa ya nama pelawaknya ya?
Jangan dikasih tahu katanya. Kayak bener.
Siapa sih pelawaknya nih kira-kira?
Disket, bukan ya? Oke, 3, 2, 1. Ada berapa orang yang betul? Komeng. Kok bisa?
13 orang loh betul.
Ada 13 orang. Kok gimana? Komeng.
KO, kan ke bawah KO.
Dan
Kucing kan meng, ya jadi komeng. 13 orang loh, betul loh, luar biasa loh. Yang bisa menebak dengan benar. Kita lihat lagi perolehan skornya.
Sius ya.
Masih ya, luar
Sius
biasa.
ini emang luar biasa. Sius pokoknya. Ya.
Berikutnya Mas Fuad.
Oh, komeng.
Iya. Komeng ya.
Cuaca sedang mendung, ada 5 orang dan 1 payung. Bagaimana cara agar mereka tidak kebasahan?
Gimana
Lari
nih
bersama,
caranya?
jalan saja, pergi satu persatu, atau menambah payung?
Gampang kan?
Mbak Nani serius banget, Mbak Nani, ya Allah. Mbak Nani itu serius.
Oh, 59 orang menjawab benar, luar biasa, mayoritas benar. Jalan aja karena kan mendung ya, belum hujan, jadi jalan aja
gitu.
santai. Betul.
Iya, betul. Karena masih mendung.
Jalan aja. Berikutnya Mas Fuad.
Apakah
Sius masih memimpin.
Masih ya. Lumayan jauh ya ini skornya. Puzzle. Urutkan tahapan memakai skincare yang benar.
Ayo, gimana nih skincare urutannya yang benar?
Ya, ini zaman sekarang Bapak Ibu udah pasti jago nih. Bapak-bapak juga banyak yang pakai skincare, saya percaya ya.
Apakah pakai moisturizer dulu baru cuci muka? Apakah cuci muka dulu? Apakah pakai serum dulu baru toner? Apakah moisturizer dulu? Kira-kira gimana?
Ini waktunya masih 25 detik lagi ya.
Tapi
berarti
sudah banyak yang submit jawaban.
Udah banyak yang bisa jawab.
Iya, keren nih. Tinggal sedikit lagi yang belum.
Ayo Sius, semangat Sius.
Sius, pertahankan posisimu Sius.
Oke. 7, 6, 5, 4, 3, 2, 1.
Berapakah yang jawab benar?
Oh, banyak ya. Facial wash dulu, toner, serum, dan moisturizer. Ya.
Siapakah yang memimpin sekarang, Mas Fuad?
Masih Sius ya.
Sofi naik ke atas ya.
Ya.
Wah, banyak nih yang naik. Berikutnya soal terakhir.
Soal terakhir. Kaluna mempunyai uang 15.000. Bila Kaluna membeli roti harganya 6.500, berapa kembaliannya?
Berapa ya kira-kira kembaliannya?
Ada foto Habib di situ ya.
Ya, jualan, jaga warung. Belinya di warung Habib. Bener ini jawabannya Mbak Nani. Bener ini serius. Serius banget ini jawabannya. Oh, masih ada yang terkecoh ya.
Jawabannya 3.500.
Uangnya 10.000. Enggak kan enggak ngasih 15.000 kalau nanya kan 10.000
Oh, karena
pecahannya.
15.000 yang dikasihnya kan 10.000, bukan 15.000. Oh iya betul.
Kan?
Ngapain 5.000-nya dikasih?
Ngapain kan?
Oh iya betul.
Oke, kita lihat siapakah pemenang dari kuis ini.
Iya.
Yang menempati podium. Yang ketiga
Nadia juara 3.
Juara 2
Dengan
Nae.
username Nay.
Juara
Dan
1
satu lagi siapa?
Siapakah? Selamat kepada pemenang. Yang menang boleh menghubungi panitia ya.
Coba
boleh menghubungi
mungkin
saya.
di sini ada yang
di sini?
username-nya disebut di depan.
Sius, ada Sius?
Mbak Rina Sius. Apa tuh?
Sius.
Nae. Oh, ini Ibu Sius? Oh, Masya Allah. Tepuk tangan dulu dong, Ibu. Hebat sekali, Ibu.
Luar biasa.
Ya. Dan Ibu Nadia? Oh, Ibu Nadia. Kita beri tepuk tangan juga kepada Ibu Nadia.
Luar biasa. Selamat kepada para pemenang. Menghubungi saya ya selanjutnya. Nanti akan mendapatkan hadiah yang menarik. Baik, terima kasih. Mungkin selanjutnya akan dipandu kembali oleh Mbak Echa. Terima kasih Mbak Echa.
Oke, sama-sama Bu Diana. Oke. Kita beri tepuk tangan dulu buat Bu Diana. Seru banget Bu Diana kuisnya. Iya. Baiklah Bapak Ibu yang kami hormati. Selanjutnya workshop sesi ketiga akan dilanjutkan dengan materi dengan judul Non-Clinical and Clinical Development Oversight of ATMPs yang akan disampaikan oleh narasumber dari European Medicine Agency, EMA, dan akan dilanjutkan dengan materi
GMP Compliance for ATMP, Production and Process Flow yang akan disampaikan oleh narasumber dari Miltenyi Biotech.
Sesi workshop selanjutnya akan dipandu oleh moderator Ketua Tim Kerja Pengawasan Produksi Produk Biologi, Dr. Santi Marlina, SSI Apoteker MSC. Untuk itu, kepada Ibu Santi Marlina, waktu dan tempat kami persilakan. Kita sambut dengan tepuk tangan Bapak Ibu.
Baik, Bapak Ibu sekalian, Assalamualaikum warahmatullahi wabarakatuh. Gimana kahutnya seru ya? Lebih milih apa pertanyaan tentang jin terapi apa pertanyaan tentang ini kodok apa buaya? Buaya yang sudah bermutant. Oke, Bapak Ibu sekalian, ada dua narasumber yang akan saya moderatori, yaitu dari satu dari EMA, ya, dan satu lagi dari praktisi. Jadi memang nanti kita buat dua sesi yang terpisah karena memang apa namanya naturnya beda ya, dan juga Bapak Ibu bisa mengharapkan bahwa nanti interestnya terhadap pertanyaan-pertanyaannya juga berbeda. Oke, mungkin kita cek dulu apakah narasumber dari EMA sudah bergabung dengan kita di Zoom saat ini. Kalau bisa di apa namanya di pin ya, Bapak Ibu di Zoom-nya. Ya, oke. Here, it's already present Dr. Patrick Salis. Dr. Patrick Salis, can you hear us?
I can hear you very well. And can you
Okay,
hear
good
me?
morning from there.
Good.
Is it in the morning? Yeah.
It's
In the
morning. Yes, it's 8:45.
Okay. So here we are already in a group of people consists of the academia, regulator, and also industrial practice. So you can expect the variation of audience. So Dr. Patrick Sals, allow me to introduce you before you start your session. Yeah. So yeah, ladies and gentlemen, please welcome our distinguished speaker, Patrick Salis, who has been in with the EMA since 1997. Dr. Salis holds a degree in pharmacy and PhD in pharmaceutical science from the University of Leuven, Belgium. As a lead scientific officer in EMA Office of Advanced Therapies and Hemato-Oncology, he plays a pivotal role as a scientific leader to the Committee for Advanced Therapies yang kita kenal dengan CAT. Dr. Sals has held several key positions within the EMA, including coordinating EU authorization procedure for biological and biotech products. His extensive expertise has been instrumental in scientific projects such as pandemic influenza vaccines, transmissible polio for encephalopathies, viral safety of recombinant products, and cell-based medicinal products. He also served as the scientific secretary of both the Vaccine Working Party and the Cell-Based Product Working Party. So here some more. So Dr. Sal have been heavily involved in implementing the regulation on ATMP at EMA. Yeah. Prior to his work, he has served as a pharmaceutical assessor at the Belgian Ministry of Health. And we are honored to have him share his valuable insights, with us today. Today, Dr. Sell will present on the non-clinical and clinical development oversight of ATMPs, particularly within the EMA's Office of Advanced Therapy. He will provide critical insight into the regulatory landscape surrounding of ATMPs. His presentation will dive into the essential elements of non-clinical and clinical development, emphasizing the rigorous oversight process that ensure the safety, efficacy, and quality of these innovative therapies. So we're excited to hear from you, Dr. Salis. Please join me to welcoming Dr. Patrick Salis. Floor is yours.
Okay, many thanks for the kind introduction. So let me just share my screen and put it in full mode. Okay, one moment. Okay, I think you can see the full screen now. Okay, so I can already skip a couple of the first slides. So my talk is officially I will be talking, or main part of my talk will be on non-clinical and clinical requirements for ATMPs. But I will delve a little bit deeper also in the regulatory framework we have in Europe for ATMPs, how to get products on the market in the EU. And then I give you a bit of insight on authorized products, the pipeline, and challenges we see with ATMPs during development and critical issues that appear during the assessment of the marketing authorization application for ATMPs. And then I will delve into the second part of my talk on the non-clinical and clinical requirements for ATMPs. So this is a quite busy slide and you don't have to remember anything here, but what I just want to highlight is that in Europe, we have in the middle here two main legislations who regulate the authorization of medicines in Europe. And then surrounding this, you have a lot of satellite legislation, and the advanced therapy legislation is one of those. The specific piece of legislation for ATMPs is a regulation that was so legal text that was adopted in 2007 and became operational end of 2008. Now, this is a relatively limited piece of legislation insofar that it's what's called a lex specialis. So it refers to everything that is in the standard pharmaceutical legislation, except for what is specific for ATMPs. So ATMPs are, as you know, cell therapy, medicinal products, gene therapy, medicinal products, and tissue engineered products. What you find in this piece of legislation is definitions on what are ATMPs, and also clarifying that in Europe all type of ATMPs are medicines and that they will be authorized via a centralized procedure, so meaning a single authorization for the entire EU. The legislation describes it describes that the principle of existing legislation for medicines will apply when it comes to marketing authorization based on a demonstration of quality, safety, and efficacy. It requires GMP and GCP for ATMPs, even though it says it needs to be adapted, and I'll come back to that in a moment. Talks about post-authorization vigilance and risk management planning, traceability, and then it sets up a specialist committee, and this is the Committee for Advanced Therapies, taking into account the specific expertise that is needed to review, to evaluate marketing authorization applications for ATMPs. And last, but certainly not least, it foresees incentives for ATMP developers. And this was the idea that we're talking about legislation that was prepared almost 20 years ago. The intention there was to support the ATMP developers, which are novel and might need additional help from the regulators to get their products approved. Before I go further, just to clarify that this piece of legislation is specific for the approval of a marketing authorization for ATMP, so for the licensing. It doesn't talk about clinical trials, because for clinical trials, the same legislation as for any other medicine apply. Now, what does the Committee for Advanced Therapies do? As main tasks, they have the their role in evaluation of authorizations, marketing authorization applications for ATMPs. And then there are two ATMP-specific procedures, certification procedure and classification procedure. In the certification procedure, the committee will look at data, early data generated by the developer, and they will, and it's restricted to quality and non-clinical data only, not the clinical data. And they will make an evaluation if the data set the developer has generated is or is not suitable to the standards of future marketing authorization. The classification procedure is one where a developer can ask if his product is or is not fitting within the definitions of an ATMP, and as such provides the developer with some legal and regulatory certainty on their product. So based on the outcome, they will know what guidance to follow and in what legal framework they have to develop their product. Committee is also involved in scientific advices, support to other committees, and is also responsible for the development of scientific guidance for ATMPs. Now, how gets a product approved in Europe? So after a developer has produced data in a clinical setting, of course, first non-clinical testing, quality and non-clinical testing, and then clinical trials, he submits a request for marketing authorization directly to the European Medicines Agency. And this is because for ATMPs, like for a couple of other products, the approval is central in Europe. Then two committees will be at the center of the scientific evaluation. So that is the Committee for Advanced Therapies and the Committee for Medicinal Products for Human Use, CHMP, and this is our main scientific committee for all medicines. The Committee for Pharmacovigilance and Risk Management, so the PRAC, will also support the evaluation of the initial marketing authorization. At the end of the process, both committees will come to a conclusion on the benefit risk, and if the benefit risk profile is positive, this will result in an approval of a product. Can be a full approval, a conditional approval in case that the data package is not complete, or an approval under exceptional circumstances. And after this approval, the marketing authorization holder at that time will have to consult with the reimbursement bodies to get reimbursement for their product. And then once the product is on the market, our Committee for Pharmacovigilance and Risk Management will then review all the post-approval data, and if there are signals that need a change to the marketing authorization, a variation, they will report that back to CAT and CHMP. This schematic just shows you the approved advanced therapies in Europe. The graph is for the last 10 years. And what you see here is that the majority of the products that are approved in Europe are gene therapies. That's the blue box in the middle. Majority of those are for orphan diseases. We have a couple of tissue engineered products and somatic cell therapy products approved, but the majority is indeed gene therapies. And what you also see is that it's especially the last 4 or 5 years that we have approved quite a lot of products. So in the first 5 years, there were 4 approvals just for completeness, and that is not shown on the graph here. The pipeline or the prediction of the number of applications for ATMPs per year is approximately 10 per year. However, what we have, and that was a prediction we made before the COVID pandemic, what we see is that there is certainly that the COVID-19 had an impact on the running of clinical trials. So we see a reduction. And what is also typical for these products is that they have a high attrition rate, meaning that products that are announced, a larger part than other medicines will not be submitted because often because of failed clinical results. What you see in the graph at the left is the number of scientific advice requests we have been looking at for ATMPs. And from 2009, when CAT started, until now, what you see is the increase of scientific advices, and then parallel to this is the number of products, ATMPs, under development and in clinical development. And especially also the gene therapies, the red bars that are now the majority of the request for scientific advice. And something similar we see in the PRIME procedure. And PRIME is a scheme that is run by the European Medicines Agency to enhance the support of development of medicines that target unmet medical needs. And through this PRIME system, the agency offers early and proactive support to medicines developers to optimize the generation of robust data, and as such, hopefully enables accelerated access of the products to the patients. So what we see here is the number of PRIME applications to the left that are submitted, and the red ones are ATMPs, and the number of PRIME applications that have granted this special additional support. And what is obvious here is that the red bar being ATMPs, that about half of the PRIME products are ATMPs. So these two slides show you that there is quite a substantial pipeline of ATMPs under development in Europe and worldwide, because nowadays the development is not really restricted anymore to any region or country. Now I move on and I give you a little bit of insight on an analysis that colleagues of the agency have done looking at scientific advice requests. And then the main points for which scientific advice requests have been asked are in the quality development on comparability and control strategy, and that was out of the analysis, 2/3 of the developers asked for questions on comparability. Then questions on toxicity and how to bridge to the clinical situation. And this is because of the specificity of ATMPs, that it is not always so easy to come up with an appropriate non-clinical development plan. And that is the second part of my talk. And then a lot of scientific advice requests on the clinical development. And this is often these ATMPs are developed for small patient populations. So that makes that a standard clinical development, like what you have for, let's say, a chemical product with a phase 1, phase 2, and then a pivotal phase 3 trial, is not something that is often seen, and for small population, not even really possible. And just to give you the example of one of the approved products, product is called Strimvelis for for a genetic disorder called ADA-SCID. For that approval, we only had clinical data on 14 patients. Now that of course have an impact on how the study design should look like, and especially how you can contextualize the data that you have, because a randomized controlled trial is hardly impossible when you have such a small population. And for the product I mentioned, these were single-arm trials that have been conducted, or a single-arm trial that has been conducted. So as randomized controlled trials are not what we standardly see, we of course will be looking at innovative designs and real-world evidence data to contextualize these single-arm trials with regards to the patient population. The next couple of slides are the critical issues that we see during the evaluation of ATMPs. So this is not the scientific advice like in the previous slide, but the real issues we see when we looked at the 27 applications received so far. First, on the quality development. So what we have there is, and the major shortcomings are highlighted here. What we have first there is issues with comparability. And this is the most frequent, what we call major objection, so blocking point during evaluation. It's very challenging to address comparability retrospectively, and frequently the analytical comparability cannot be done because the methods were not yet stabilized in early development, and or materials have expired or are no longer available. So a message to any developers is that a good comparability plan set up during development, early in development, is very critical, because otherwise your efficacy data, clinical data, can be compromised if it's not sure that your clinical batches are of appropriate or can be compared when there are changes in the manufacturing process. The second point often seen is potency, and potency is difficult because it tries to link the biological activity in terms of clinical effect to the quality characteristics of the product. So potency testing can take long, sometimes even years, to develop and validate, but it is very critical for confirmation of efficacy and comparability. So it's important that this assay is in place at the time of marketing authorization, time of approval. Ensuring product consistency is another point, and always complex for ATMPs, because there, of course, the manufacturing process is difficult, and you have a lot of variability that is coming from the starting and raw materials. And that brings me to the last point I want to highlight on major critical quality questions during approval. And this is issues with regards to the starting material, which is, of course, the starting point of the GMP manufacturer. So here the expectation is that the starting materials are thoroughly characterized and well defined. On the non-clinical questions there, it is understood, and I will talk about that in the second part of my talk, that the standard non-clinical development cannot
Dr. Salis, are you still with us?
be conducted. However, this needs to be that approaches need to stand explore. An unprofile will hamper or could hamper seriously
Bapak Ibu kayaknya koneksi ya, terganggu. Kita lagi coba reconnect ya.
Can you still hear me? Can you confirm that you can still hear me?
Yeah, Dr. Salis, yeah, probably we having a bit connection issue, but now we can, we already reconnect with you, and then you can really share your presentation again.
Okay. One moment, I'm sharing again.
Yeah, the last slide, I believe, is that still.
Yeah. So let's see, you can see the slides again. So let me now go back to where I can see it. Okay, so let's perhaps start here again. So
Dr. Salis,
yeah.
At our end, it's not yet shown your.
Yeah. Okay, I wait until. Okay, one moment. Try again. Okay.
Yeah,
Is it working now?
Yeah, yeah, perfect. Thank you.
Okay, where did you miss me? Did I?
Yeah, I think here is
Shall I go to back to the previous slide, or do you want me to
no, no,
continue?
you're good. This is the last where we yeah, this is good.
Apologies, here at the
Dr. Salis, can you hear us? So doctor, we're having a bit issue here. Just bear with us a moment. Maybe we need to fix this. Dr. Salis, can you hear me? Bapak Ibu, kita kelihatannya having a lost connection with Dr. Salis because he is not responding. Kita tunggu ya, 1-2 menit, nanti kita coba lagi. Ini Dr. Salisnya ada di room Zoom? Enggak, ya itu ya. Mungkin panitia bisa email beliau kali. Advice kalau kita lost connection. Bapak Ibu selagi menunggu mungkin bisa dicek and recheck presentasinya Dr. Salis untuk menyiapkan pertanyaan.
Mohon izin Ibu, kami dapat info di sana Dr. Selis sedang proses untuk join kembali.
Oh,
Ya.
ya udah. Kita tunggu kalau gitu ya. Oh yeah. Dr. Salis, can you hear us?
I am back now.
Okay, you may continue, but bear in mind
Uh,
because of this trouble issue, you only have like 15 minutes
yeah,
left.
I will go a little bit faster. So
Okay.
apologies for this.
No worries. Yeah, all good.
Okay, let me.
Continue.
Let's hope there's no issues this time. Share screen 2. No, that's not. Okay, I hope you can see my screen.
Yes, perfect.
Okay, so just continue and I will take it a little bit faster. Just critical aspects, I will not delve in detail. I will just mention them because I come back to that later in my presentation. So for critical non-clinical issues we see are the fact that you normally do not have a standard non-clinical development, so that needs appropriate justification and additional alternative data generation. You, of course, have lack of relevant animal and non-clinical models. That is an issue. And for certain ATMPs, there are specific safety issues that need to be looked at, and coming back to that one. For GLP, there's the requirement for the pivotal tox studies to be done under GLP, and guidance is available there. Going into the critical clinical issues is how to conduct the trial design for this type of products, taking into account that often, like I said, it's a limited data package that have been generated, very often not a randomized controlled trial. How to get the indication right. Indication not reflecting the patients treated in the clinical trial. Is the safety data package sufficient for establishing the safety profile, which is not always the case. And then a very tricky one, an important one, is the durability of response for these products. You know that ATMPs are often only given once in a lifetime, and then you want that these products are showing effectiveness over a long spell. So ideally, of course, lifelong, but certainly a long time. But you cannot study a lifelong or a 5 or a 10-year effectiveness in a clinical study. So that's where registries come into the play. So let me now go to the second part of my presentation, and this is on the dedicated specific scientific guidance we have for ATMPs. There are three main that I will highlight further on. The first one is the guideline on cell-based ATMPs. So this is the somatic cell therapy product and tissue engineered product type of products. The second is for the guideline for gene therapies. So this is a non-cell-based, vector-based type of gene therapies. And the third one is the guideline for genetically modified cells. This last one is one where you have also product-specific guidance for CAR-Ts, so both clinical and non-clinical, for induced pluripotent stem cell-derived products, non-clinical, and for genome-edited cell-based products, also non-clinical. Now, just to stress that the three guidelines that I mentioned will talk about the requirements for marketing authorization, so that we are working and are close to finalization of another guideline, and this will also be an important one to look at, and this is for clinical trials. So the requirements for ATMPs in clinical trials. And this is well understood that in a clinical trial, of course, your quality development, for example, is not yet fully there, and validation of manufacturing process and so on is not there. So this guideline under development will address these phase-specific requirements for ATMPs. Now, going further, there's quite a lot of additional guidances that are published. Guideline on risk-based approach, which will guide the non-clinical quality, non-clinical and clinical development. Then also an important one is the guideline on safety, efficacy, follow-up and risk management. And then we have quite a lot of Q&As, so more informal guidance. For example, comparability is a very important one. Like I said, the legislation also requires that ATMPs are developed under GMP and the trials are done under GCP, and that the GLP principles are there. So there is specific ATMP specific guidance developed on GMP, GCP, and GLP. And all these guidances can be found at the EMA web page, so www.EMA Europa EU. And then once you are there, I will quickly guide you. You go to the second tab, which is regulatory, human regulatory. In human regulatory, you go to pre-authorization activities, and then you go into scientific guidelines and multidisciplinary guidelines. That's the place where you find all the guidelines for gene therapies and cell therapies. Now coming to the main bit of, or the second bit of my presentation, and this is on the non-clinical and clinical developments of ATMP. So first, couple of general statements. So as you know, ATMPs are very diverse, going from a simple expanded cell product suspension to very complex genetically modified cells that incorporate a genome editing step. So therefore, the non-clinical, equally the quality and the clinical development, will not follow a one-size-fits-all approach. The risk-based approach will guide the development, quality, non-clinical and clinical, and the extent of non-clinical testing in the extent of the testing, you can take into account the specificity of the products and some prior knowledge with similar products. What you will see when you would study and will study the three guidelines and the fourth one under development is that there's certainly a move away from animal testing to rather an ex vivo, in vitro, it should be say in vitro instead of in vitro, ex vivo, or in silico models. So it's the current statement is that animal testing should only be done if it will generate robust and meaningful data. Non-clinical studies can be either standalone or combined, and a good quality CMC development is key before starting, especially starting the pivotal non-clinical testing. So be sure that your product manufacture is more or less stabilized before you do the pivotal tox study, and certainly before you start the first in human trial. Otherwise, you enter into problems where it's very difficult to link your non-clinical testing to the clinical testing and to the quality of your product. So pharmacological studies, so proof of concept studies, where the request is to use a relevant non-clinical model, which could be a humanized or an homologous model or a humanized transgenic animal model. What to look for? Proof of concept that of course depend on your mechanism of action. So for gene therapies and genetically modified cells, you are likely going to look for transgene expression and genome integration when you have an integrating vector. For tissue engineered product, you will rather be looking for restoration of function, and for somatic cell therapy product, you will depend, of course, on the indication. For example, you will look at immunological action if you have an adoptive immune therapy. Extrapolation to humans is always difficult, and it can, for example, be hampered by the different tropism of the vector, gene therapy vector in animals, or a different biodistribution, the absence of a good non-clinical model, and so on. Safety pharmacology is expected to be done or justified if not there. When it comes to pharmacokinetics, there standard absorption, distribution, metabolism, and excretion studies normally not relevant for ATMPs, but you will be looking to distribution, migration, expansion, and persistence of cells when you have a cell product. For gene therapies, you will look at the distribution profile and clearance. You will study genome integration, and this is for vectors that are supposed to integrate. You will have to have a look at germline transmission and modification. And here I want to refer to the ICH S12 guideline. And you will have to conduct shedding studies. And here as well, there is an ICH consideration document to look at. Tox studies are required to generate data, but the data should be meaningful and relevant to support the safe use of the product in the clinical indication. Conventional tox studies, not always possible. And as most ATMPs are single administration products, single dose tox with an extended observation period is what you should be looking at. Can be combined, as I already said, and ideally the dose level route of administration should be representative to the clinical use. Pivotal non-clinical tox studies, safety studies to be done in conformity with the principles of GMP. Specifically for gene therapies, you will also study unwanted consequences of biodistribution of persistence of the vector, infection, transduction, transfection issues, expression of biological activity, and unwanted pharmacological effect of the transgene or the vector genes. And case by case, you will study, should study also genotoxicity, risk of insertional mutagenesis, tumorigenicity, and reprotox, as well as immunotoxicological studies. For genetically modified cells, you will look at toxicity effects related to the cells and to the transgene expression. You look at the risk of insertional mutagenesis, that's because of the gene part, vector mobilization, and recommendation is not the correct word, reassortment. So when I go, for sake of time, I go a bit faster on the clinical. Same principles as for other medicines, but adapted to the specific characteristics of the ATMPs. Not standard clinical development. First in human are done in patients and not in healthy volunteers. So there need to be some benefit expected for the trial subjects already in the exploratory trial, even though the primary objective will still be safety and tolerability. And often it's not possible to re-administer products, so that's another argument why there should be some expected benefit already in the first-in-human trial subjects. You will look at dose finding, dose escalation, which is of course more difficult for ATMPs. So this could be that you provide a justification for absence. The confirmatory or the pivotal trials should follow as much as possible the therapeutic area guidance, ideally randomized controlled comparative trials over a single arm trial or a trial with an external or historical control, even though this is not always possible. But blinding is very important as far as this is possible. And if single or double blinding is not possible, it should be appropriately justified. And that situation could be when there is a surgical procedure versus placebo or not. And in that case, you will look at blinding of the person that is assessing the study outcomes. And because it's not possible to blind the one who is administering the product. We're looking at clinical endpoints or validated surrogate endpoints, and this is a bit of a divergence with US, who will allow for surrogate endpoints only for their conditional approval. Where in Europe, we always want some clinical endpoints as well. Clinical safety, the risk of the administration procedure need to be addressed, and also the issue on medical devices for delivery or implantation. So it need to be well demonstrated what devices are used. Duration of trial to take into account the treatment effect and late onset signals, but this is where the long-term safety and efficacy studies post-authorization or post-clinical trial become important. Let me just see if I have anything. I will just skip this. You will have the slides. For sake of time, I just want to conclude with a couple of take-home messages. So for ATMP development, we expect that the same high standards as for other medicines are applied, but of course there is tailored guidance and a risk-based approach developed for these products. Efficacy and safety starts with a good quality of the product. It is important to collect as much as that possible already at very early stages. This is because you often are dealing with small populations, so any clinical data obtained from any patient is relevant and it might become important for the approval. Important is the long-term data collection, both for efficacy and for safety in the post-authorization phase. And this is where the possibility for registries and real-world evidence comes in. And we have tools and incentives in Europe for product ATMP development, and I have listed them here. Scientific advice, very important. Early interactions with the regulator via a meeting with Innovation Task Force. The 2 ATMP specific procedures, classification and certification. PRIME as a non-ATMP specific but very important for ATMP early support mechanism. The scientific guidelines are there, and there's a possibility for additional support to small and medium-sized enterprises developing ATMPs and for academics developing ATMPs. And with this one, I conclude, and I apologize for the technical hiccup in the middle of the presentation.
Thank you very much, Dr. Salis. We need to give a big applause for his effort on sharing the experience of how Emma have assess or evaluate the products. And we actually still have 10 minutes for a question because this is a very rare opportunity. So I hope you can still stay with us, Dr.
Sure, absolutely okay.
Yeah. So anyone, I think we can actually accommodate 3 questions, maybe. Yes, yeah. Pak Gagas.
Good afternoon, Mr. Patrick Salis. Can you hear me?
Nice and clear. Please go ahead.
My name is Gagas from Badan POM or Indonesian FDA. I'd like to ask about clinical requirement for ATMPs. At your slide number 26, there is a statement, there's no standard clinical development phase, whether it's phase 1, 2, or 3. I have two questions. The first one, is there any particular reason why we can't use standard clinical development phase? And the second question is, from your experience, do you have any, do you know what is the common clinical study design for conducting experiment on ATMPs?
Yeah, I can try to address this one. The phase 1 study is a study that is conducted normally in healthy volunteers. That's but for ATMPs, there you have to take into account that these are not the standard products that will disappear from the body after a certain time. So the ones you administer a gene therapy or a cell therapy, the consequences will be that the product stays within the human body for a long time, if not lifelong. So studies in healthy volunteers is not something that will be routinely done. In all the products and scientific advice I have seen, I've not seen any study proposed in healthy volunteers, and likely the ethical aspects of approval of a clinical trial in healthy volunteers with a gene therapy or a cell therapy are not allowing for this one. So that's the first thing. So what we normally see is a combined phase 1, phase 2 study, where you will have the characteristics of the phase 1 looking only at safety tolerability, but already have some secondary endpoints addressing the clinical use, clinical efficacy of the product in patients. So that is what we normally see. And then we see either a phase 2-3 study as the pivotal study or a real phase 3. So that depends a little bit on the type of study design and so on. So what we normally would like to see is, and we're trying to push the developers for it, is to do some kind of randomization of the pivotal study, of the phase 2, 3, or the phase 3 trial. And in some cases, it should be possible, even though most of the studies that we see so far are single-arm trials. Single-arm trials with then an external control or an historical control. So that is what we see often. The arguments for the single-arm trial are valid and not valid. Of course, if you have my example of a trial where you have only 10 patients or 20 patients in your clinical development, it's not that easy to do a randomized control trial with sufficient power in such a limited population. Additionally, it might, especially for very orphan indications, not be that easy to do a randomization because you don't have any other standard of care. So it's very difficult to do a randomization against an, let's take again the Strimvelis case, randomization against you will give some patients the gene therapy product versus you will give some patients the bone marrow transplant, hematopoietic stem cell transplant, which is the other indication. So that is difficult to do a trial over two type of regulatory frameworks, but might be possible in some cases. And we are certainly pushing for products where there is a larger patient population like CAR-Ts to for the developer to do a clinical trial versus standard of care. So randomized trial versus standard of care. So just to recap, we are flexible when it comes to the phasing of the studies, where we look at an exploratory versus a confirmatory trial as the basis for approval. We are trying to push for randomized control trials, even though routinely we see now single-arm trials with an external control. I hope that addresses a bit your question.
Thank you, sir.
Pak Gagas sudah cukup jelas. Yeah, we can actually take one more question. Maybe Bapak Ibu sekalian silakan, ini sangat jarang dan kita kan juga belum punya banyak pengalaman untuk mengevaluasi ATMP ya. Jadi pergunakan kesempatan. Yeah, there is one more question, Dr. Salik.
Yes, hello, Doc. I'm Hikmawati from B Pharma di Luhung. So I would like to know regarding the comparative drugs which can be used for the clinical and non-clinical. As you know, maybe for the ATMPs or actually our company is developing for secretum, which is there is no country that have been registered this product also. So we are confused to choose the comparative drugs for this product, and there is some discussion actually between our researcher to use such as hyaluronic acid, which is it is classified as medical device. If we choose that for the comparative, is it apple to apple for being used as comparative with our product? And yeah, and
Okay.
maybe have a suggestion for the non-clinical also for the comparative drugs. Thank you.
Yeah, it's apologies. It's a bit of a difficult question because it's already going product specific. So what we normally would say is that, going back to a CAR-T product, it's very difficult to ask for a comparative trial against another CAR-T product because these are not products that are routinely available and used as available as a comparator in a randomized control trial. That's why we often refer to standard of care as the comparator arm for a clinical trial. If for your indication, hyaluronic acid would be a standard of care, I would say that is a potential good comparator in the study. Now, like I said, this is product specific, and what we normally would advise any developer in such case is to ask for a scientific advice to your authority and put that question to them and see if they agree with your argument to use hyaluronic acid as a comparator in your trial. Now, for non-clinical testing, you normally do not have comparative studies, at least not that I remember ever seeing in non-clinical a comparative study. Perhaps you want to elaborate a little bit more what you mean on comparative study and non-clinical.
So actually for the non-clinical, we also confused to choose what is the comparative drugs, because the comparative in non-clinical or preclinical, it is have to align with the clinical trial design, right?
Yeah, I don't think that in Europe we would ask for for that. We will, of course, try and put in the guidance that the non-clinical route of administration and the non-clinical product should be as close as possible to the one that is used in the clinical trials. So that is something we see. But we will not ask studies with a comparator product, non-clinical studies with a comparator product.
Okay, okay. So we don't have to
choose any comparative drugs for the preclinical, but
No, normally not. You study the toxicity, the proof of concept, toxicity profile of your product under development and not of the comparator product, because that is something that should have been done by the other developer, the one of the comparator product.
okay. Thank you, Doc.
Ada lagi ya? yang
Mohon
mau
izin,
bertanya?
Ibu.
Iya.
We have one question from the participant who joined via Zoom. Do we still have time?
Yeah, it's okay.
Okay.
We can have a 10 minutes delay. Dr. Ulf, can you
Yeah,
just
that's all okay for
generous
me.
to,
Yeah.
yeah.
Okay, from Nana, from Indonesian FDA. The question is, in what cases do we need non-clinical assessment of tumorogenicity for gene therapy? Maybe I will repeat. In what cases do we
Yeah.
need non-clinical assessment of tumorogenicity for gene therapy?
Okay, so normally you would test, and that's how it's put in the guidelines, you will test for tumorigenicity or in a case of integrating gene therapy vectors. So this is the standard requirement. So if you have a lentiviral or a retroviral vector that you are using to transduce cells, then you would indeed look for the risk of integration and the risk of tumorigenic potential due to the integration of the vector. I have to say that there is a bit of a moving field insofar that the novel technologies, genome editing in specific, could raise also the potential of tumorigenicity, and at least the applicant will have to address the risk of tumorigenicity due to gene editing technologies. When it comes to non-integrating vectors like the AAV, so adeno-associated viral vectors, there the situation is not that clear. And that's a bit of a moving target for the moment. Where we are aware that there is, that is relatively novel information, that there is a limited risk, there is a risk of integration of these AAV vectors. So hence there could also be a risk of tumorigenicity. However, the risk there is much lower because the frequency of integration of these AAV vectors is also lower. So for that one, we would probably still accept that the company comes with a good scientific justification for not doing tumorigenicity investigations instead of starting some testing of tumorigenicity. So so to recap, integrating vectors, yes. For genome editing, likely. For AAVs, potentially okay when there is a good scientific justification for not doing it. That's the current thinking, but this is a bit of a moving field.
Oke, ya, saya rasa sudah cukup ya Bapak Ibu. Kita sudah 10 menit ya, sekitar behind the schedule. But Dr. Salis, thank you very much again for sharing with us. This is a very valuable and fruitful sharing, and we would like to thank again. And I hope that in the future. Jadi Bapak Ibu sekalian, berikan sekali lagi applause buat Dr. Salis.
Mohon izin, Ibu. Before we ending this session, thank you, Dr. Serif, for your excellent presentation. In recognition of Dr. Salis' outstanding contribution, we would like to present him with a certificate of appreciation. Please give a round of applause.
Okay. So thank you very much for inviting me, and I wish you a further successful session and meeting for the rest of your day and days. Thank you.
Enjoy the rest of your day.
Thank you very much.
Ya, jadi Bapak Ibu sekalian, kita akan lanjut ya untuk sesi berikutnya yang juga akan dilakukan secara Zoom. Kita akan lihat dulu apakah narasumber untuk sesi ini sudah hadir di ruangan.
Sudah.
Okay. So, I am excited to introduce our next speaker, Dr. Betke. I hope this is already like so many couple times.
Very good, very good.
To practice how to pronounce your name. So apologize if it is not really accurate, but Dr. Betke, is that correct?
This is
How are you,
correct.
doctor?
You can hear me?
Yes, we can actually hear you. And I can see here that you bringing also your colleague from Miltenyi. So we are very happy for you and your team to join.
Good. Yeah.
So allow me to introduce you, doctor, for bringing you to the next session. So here I have that Dr. Ulf Betke is an executive expert in cell and gene therapy for the APAC region at Miltenyi Biotech BV and Co. KG. Since 2022, Dr. Betke has been instrumental in supporting the implementation of manufacturing cell-based therapeutic products with a particular emphasis on Clinimax Prodigy-based CAR T cells. His extensive experience includes clean room design for cell-based manufacturing process, GMP manufacturing, and quality control, all aimed at achieving manufacturing permissions and regulatory approval for point-of-care and in-hospital solutions. Dr. Ulf holds a doctorate in biochemistry and immunology and is a specialized pharmacist in pharmaceutical technology. His career began at Biotest Pharma in Frankfurt. He established a manufacturing plant for therapeutic monoclonal antibodies and led a development of a humanized interleukin-2 receptor antibody. In 1994, he joined Miltenyi Biotech, established the Clinimax system in collaboration with Amgen Corp, which received CE certification in 1997. As the head of technical director of the clinical product division, Dr. Ulf oversaw clinical manufacturing, quality control, QA, and regulatory departments, growing his team from 7 to over 350 members and supporting sales exceeding 250 million euro. So today, Dr. Ulf will present on the GMP compliance for ATMPs, production and process flow. So please join me in giving a warm virtual welcome to Dr. Ulf Betke.
Thank you. I have a little bit difficulty to show my presentation. Can you see my presentation?
Yes, just maybe a couple of seconds until it's fully shown in our screen. This is only a quarter of it, Dr. Ulf, so you may want to fix it.
I'm not sure what I have to do. 1 second. Neue Freigabe. Can you see it now?
Not yet, Doctor.
What is
screen?
I'm trained in Teams, but not in Zoom.
Do you want us to share on behalf of you instead?
Bildschirm. 1 second. I try to test it again. Diesen Bildschirm teilen. 1
I
second.
think it's coming now. Let's see.
Can you see it now? No. I don't know why it's not working. I see my screen, but otherwise, what can we
Do you
do?
want us
I don't know.
to share the screen so that you
Yeah,
can
I
just
think it is the
notify
best
us
way
whenever you
to
want?
be pragmatic. I don't know why it's not showing my presentation. I have no idea.
This is now okay.
Good.
PDF
Okay,
format.
but you want to see my slides. This is the problem. I have no idea why he is not showing my. No, I'm not abbrechen. Okay. I'm a little bit under stress why he is not showing my slide in Zoom.
So Dr. Ulf, we can
Yeah,
actually help you with the sharing the screen if you don't mind.
okay. Can you see it now? No, ne? Derzeit geteilt. You see my screen, but I have no idea what is not working. Okay, then can you? I sent you the presentation. Then I will use your slides. I think this is the easiest way now. I'm sorry, and I have to apologize that it is not working. I don't know what you are seeing. This is my problem.
This is only a half quarter of your screen there. So yeah, it is not readable. Can we try to share and then you probably.
Okay. What can you see? I don't. I really have no idea why he is not showing my presentation. Do you see it now? No, no.
No, this is still the same, doctor. It's still just a quarter of the screen. Coba. Yeah. Do you use the extension of screen extension? Maybe you want to disconnect the screen.
Can you hear me?
Yes, doctor. Yeah, we have your PDF files here. If you want, and then you can.
Yeah, try your take my PDF file. It is not. I don't know why he is not showing my screen. I have never experienced this before. It should show the presentation, but it's not doing. No. Okay, good. Yeah, take my PDF on the screen and I will follow the PDF. Yeah?
Okay, so we share the screen and then you will follow.
Yes. Please.
Doctor. Okay, yeah.
I don't know what's
Try
happening.
to stop share now your screen and then let us do this the sharing.
Good. I stopped my video. Okay.
Yes.
Okay, thank you. So I would thank you very much for the introduction of me and our work at Miltenny. And as you can see, we started to work with stem cells over 30 years ago. And the experience working with stem cells and isolating them for therapeutic applications yield a lot of other clinical support products. Next slide. And this is the agenda. I would like to present you some market data. Then I would share with you our experiences in the GMP and design control of cell and gene therapeutic products, especially with the emphasis on CAR T cells. And last but not least, I would like you to share with the new regulatory developments in Europe, which Dr. Sillis already attached, and I want to share it with you because it has important implications for point-of-care applications. Next slide. This is a short, brief overview about Miltenny Biotech. We have now over 4,000 people worldwide. We are supporting R&D activities, and many R&D activities resulted in products to enable clinical manufacturing of cells and to do the necessary quality control of cells. And working with so many customers and so many countries and their regulatory bodies, we gained a lot of experience to support our customers and hospitals and industrial partners to develop cell-based products. Next slide. Here's our locations in Asia. We have training centers in Singapore and in Shanghai, where we invite interested customers, industrial partners to come to our facilities and be trained on different methods and instrumentations. And we are also, especially in Singapore, are working closely with the Viva Foundation to enable clinical training for doctors and physicians, how to treat patients in certain diseases, stem cells, CAR T cells, other ATMP products. Next slide. And this is more related to the work of my team, as we started with CD34 selection in 1997, and all these experience resulted in the design of the Prodigy instrument 15 years later. And this is what I would like to talk in the next slides more intensively on it. Next slide. The actual, and I would like to talk about the market, just to give you an idea about the dynamic. Next slide. The medical needs for CAR T cells are very high. DBCL and multiple myelomas are over 240,000 cases every year. And also, interestingly, autoimmune diseases like multiple sclerosis and lupus are now becoming target of CAR T cell therapies. The actual situation is driven by the fact that the manufacturing capacities for CAR T cells are in the range of 6,000 to 9,000 products a year, mainly by the big players, Novartis, Gilead, Bristol Myers Squibb, and Janssen Pharmaceuticals, and that the emerging markets are not targeted by the big pharmas at the moment. So there is a lot of medical need. And the next slide, please. This is just to give you an idea about the market size. We have 6 registered CAR T cell products, 2 for multiple myeloma, 4 for CD19 based against DBCL. And the most prominent product is the J&J, is the Jakarta product of Juno Gilead, followed by Breyanzi. Kymriah is not so. Novartis Kymriah product, the first registered CAR product in the world, is decreasing. There seems the effect that Jakarta and Breyanzi are more stronger in the treatment. This should you just give you some ideas about the market size, which is actually happening in Europe and United States. Quite interesting is also that B-specific antibodies, so to say, are developed in parallel to target the same diseases, and they are also quite successful. So the overall clinical field is under a lot of dynamic to develop the real therapeutic regimens. Next slide. So now talking about the markets, I would like to draw your attention to GMP background and how to manufacture CAR T cell products. Next slide. I'm sure you all know how to manufacture a CAR T cell product, taking the patient T cells and then introducing the gene and then cultivating and expanding and then freezing or finally formulated. And the overall process is quite cumbersome. It contains a lot of open handling steps, and this bears quite significant risks in the manufacturing, and especially in the scale-up of the manufacturing. So even every open handling step is matter of sterility issues, is matter of chain of identity, follow the line issues. You don't want to have mix-ups. And every open handling step is a manual process, not done by operators, and you need a lot of trained operators to do this. Nevertheless, the pharmaceutical industry achieved to manufacture several thousand products by such processes. I just want to give you one example. Close to Amsterdam in Europe, Kite has established a facility to manufacture about 1,400 products a year, maybe even more. The data are 2 years old. And 800 people are working there, and they are doing a very, very great job. But this job is very hard to do because you have to take care of so many things. Especially clean room requirements you need for open handling steps, Class B clean rooms. And so it is possible to manufacture this way, but it's very difficult. Next slide. Such CAR T-cell facilities with open handling steps require, so to say, segregation of each, so to say, patient product. And such facilities, you can see it from this picture, are very hard to design, hard to maintain. And therefore, next slide. Next slide. One target is to optimize the entire product process and to bring open handling steps to a closed system with less operator interaction. And such system is the Clinimex Prodigy, which we developed based on the experiences we had with stem cells. The initial, so to say, user requirement for such an instrument came from the regenerative medicine. Doctors wanted to use stem cells. They want to separate stem cells during a heart surgery, bypass surgery, in order to have stem cells of the patients to regenerate scarred heart tissue. And this was the initial idea, 2012, to develop such a machine based on the original Clinimex Plus. And then the dynamic of CAR T cells became, so to say, self-dynamic, and we developed the Prodigy to enable a complete closed manufacturing workflow to manufacture CAR T cells, but also other cell types. What you can see here on the picture is a typical picture of T cells in the culture. Activated T cells are growing in little clumps and clusters, showing that they are healthy and well-doing. And the entire system requires less user interaction, and it is mainly based on tube-tube seal to ensure sterile connections. Next slide. No, other way. Yes. So having such a closed system available moves the entire manufacturing from Class B clean rooms into Class C clean rooms. And this is much easier to maintain and to operate. We and our customers we are consulting with several authorities worldwide. And I summarized here our experience. So Class C clean rooms for Prodigy, as you can see on the right column of the table, is established in the US and the EU and China, Korea, Japan, and other countries like Thailand. And the major reason is that it's defined as a functionally closed system. And then based on this prerequisite, you can operate the Prodigy system under Class C clean room requirements, and you only need 20 air changes per hour instead of 60 air changes per hour. You don't need separate people to control for microbiology contaminations, etc. So overall, clean room costs are only about 1/3 to 1/4 of a Class C clean room than compared with a Class B clean room. This is not the closed system is not the only advantage of the Prodigy system. As you know, it is providing an automated process, and the automation is reducing the user interaction, and by this reducing the risk of, so to say, personal errors. And the reproducibility of the process you are running is much higher than with processes which are open and which need a lot of user interactions. Next slide. We were often asked by authorities is our system really closed? And we did a lot of different tests, which I wanted to show you on the next two slides, that how we validated the functionally closed system. We did these bioseal test on the filter of the culture chamber, of the cultivation chamber. Next slide. And we did also bacterial challenges test, where we sucked the entire disposable tubing set into bacterial suspensions in order to show that the system is closed. And this is what we validated, and which is mainly asked by all health authorities. If you are using such a system with a disposable tubing set, you have to validate the integrity, and our users need to do aseptic process validations. That means they simulate the process with peptone solution, bacterial growth solutions, in order to show that the system is kept closed. We, in our test systems, kept the system for 50 days with peptone without showing any bacterial growth. And having done this, next slide, having done this and shown this, the Clinimex Prodigy Ballroom facility, it does possess a very simple design, which is shown in principle on this slide here. The following slide shows more the characteristic. Everything is moved to a closed system, and then you can design the facility in a very clear and open fashion. And I show you on the right side the example of the British company Autolus, which are working with us, a picture of their facility. And it was very quickly done. From 21 to 23, they did all the facility validation in order to, with the Prodigy system, in order to run their clinical trials. Next slide. This is a picture of our Bergisch Gladbach facility in Germany, where we run 2 clean rooms, Class C, with a total of 16 Prodigies. And we received a manufacturing permission in 2018. And this manufacturing permission is a generic manufacturing permission. That means it is a manufacturing permission for CAR T cells, lentiviral vector transduced. So we can manufacture different CAR T cell type as long as the process stays in principle the same, lentiviral vector transduced process. And this enables the use of, this is accelerating the clinical development. So under clinical development conditions, we can use several different viral vectors. Now one process is moving into phase 3 and commercial, and under phase 3 and commercial conditions, you have, so to say, separate clean rooms for separate products. But you have still, because we are talking about an autologous product, you have still several patients in one room, which is possible because we are working with a functionally closed system. Next slide. This is the example of our facility in San Jose in the US. And next slide. This shows the principle of the ballroom design. We go from, so to say, from south to north. We have supply unit. You have material and personal airlocks. Then you have the prodigy room in class C. And this is, so to say, a very simple but schematic design for a clean room where you manufacture for clinical trials or point of care, and where you manufacture limited number of CAR T cells. The, so to say, commercial way to do it is shown on the right side, where you design a facility which has only a one-way path. People, product, waste, raw materials only go into one way in order to reduce the risk of mix-up, having a clear chain of identity. And such a facility, next slide, we have designed in Gaithersburg in Maryland. What you see here is, so to say, on the picture showing the, so to say, empty space before we installed the clean rooms. Then you see the central hallway of the clean rooms, for the clean rooms, and on the right side is the schematic drawing of three commercial suites, and in green, and three clinical suites in pink, for the entire of the facility, which is now US FDA approved. The entire, I can go in more details if we have more time. The entire workflow and people flow is from the north to the south. In the north, you see personal logs, receiving areas. On the right side, or next to the green, on the right side is the complete logistic area for freezing, packaging, storage, maintenance. And on the south side, we have the entire quality control area. And the facility is operating now. We started with clinical manufacturing, and we will go with one big car manufacturing also into the commercial suites and phases. Next slide. This is, so to say, summarizing the difficulties, just for your overview, to manufacture patient-specific drug products and the challenges which we faced. The major point is one batch is one patient product, and this results into a high quality control demand. Yeah, because you have to do so many quality control testings. I don't want to go into details. You have the slides. Next slide. What I want to point out, and which is, so to say, following the recommendations of Dr. Zelis, is the question from how do I manage the transfer from early development to commercial manufacturing? Or the other question, different, is how do I make sure that the product which I tested in the early phases of the development is the same as I will manufacture during phase 2, phase 3, and commercial phases. So the entire pharmaceutical development of ATMPs can be seen as continuous translational effort, and to show and to prove comparability of each of products from early development when you do your potential animal preclinical studies towards commercial manufacturing. And commercial manufacturing always goes in hand with transfer to a second and third or fourth manufacturing site, because you will not have one manufacturing site worldwide. And one big example, one big advantage of the Prodigy system is when you work with such a system, you are using the same system in early development up till commercial. So the use of such systems is reducing the demands in comparability comparison efforts. But the question of comparability is based on our internal knowledge for our own product, but also in discussions with our Prodigy customers. The comparability question is one of the key questions in ATMP development, in the pharmaceutical development of ATMPs. And you have to use quality of design methodology to show this and to ease in your comparability proof. Next slide. Yeah, this is, so to say, summarizing what I just showed before. You go from process development to commercial phase, and you have to have a clear idea what are your quality parameters, what are your critical quality attributes, what are your critical process parameters, how dependent is your process on raw materials, on changes of raw materials, on raw material vendors, and these questions result into a quality by design approach, a risk-based approach, where you identify the risks of which factors. The question is which factors might influence the quality of my final cell product. What can have influences? Process times, medias, serums, cytokines, the raw material, the starting material itself. Each patient product is a different starting material, how to standardize this, lentiviral or other genetic vectors. Okay, next slide. And all these questions resulted into, I want to share it with you, the next generation manufacturing strategies. To go into details is a separate product, separate talk, but we are seeing now accelerated cell manufacturing. Novartis published a 2-day process called T-Charge. AstraZeneca is working with a Chinese company on 1 to 2 day processes. This has advantages and disadvantages we can discuss. Process automation is clear, a high demand if you want to scale up. And in animals, the in vivo cell manufacturing, meaning doing directly transfer with viral or other vectors into the patient to generate CAR T cells, is under active evaluation. Next slide. I want to share with you some ideas on the transduction which we are and our customers are working on. Our major vector is a lentiviral vector, which is targeting the LDL receptor, and the LDL receptor is expressed on dividing cells. So the lentiviral vector is now is established as a safe transduction method. HIV patients never developed a lentiviral generated tumor, so lentiviral vectors are accepted as very safe method to do transduction. We have also the limitation is it's only going into rapidly dividing cells, but using a baboon so-called pseudotype vector, which is targeting an amino acid receptor. We can also target, so to say, non-dividing cells. Gamma retroviral vectors are discussed to also induce tumors, and piggyback messenger RNA and CRISPR-Cas methods are under development, but researchers and clinical developers need to do several safety measures, which I want to share with you on the next slide, please. The piggyback, as you know, is a plasmid-based transduction method, but it was causing, in these published trials, 2 of 10 patients developed secondary lymphomas. A detailed analysis shows that over 50 vector copy numbers were generated into the genome, and this resulted in the US FDA requirement that the vector copy number of 5 should not be exceeded. But it is very difficult to control vector copy numbers using standard transfection or electroporation method with plasmids. So researchers and clinicians have to work on it to improve the piggyback or other transposome systems. Next slide. And then last thing I would like to mention is the fresh or cryopreserved CAR T cells. We know that every manipulation of cells is every manipulation of the cell generates an epigenetic memory, and we don't understand everything now, but we know that every severe manipulation has a epigenetic memory. We don't understand the consequences, but if you look for these from Nirav Shah published data, it shows that you need much more, so to say, frozen cells to generate an effective CAR T cell response than a fresh cell, non-frozen cells. And these data speak for, so to say, on-site point of care in hospital manufacturing method to avoid secondary manipulations of cells. Next slide. So everybody is talking about manufacturing, but the real task is quality control. Next slide. Because quality control and quality assurance cover over nearly 75% of all your activities when you manufacture CAR T cells. These are data of our own facility, manufacturing about 200 patient products in a 12-day process per year. So this is what we calculated the time. The manufacturing is limited 20%. And but the major task is quality control, and therefore we have to automate it to enable large-scale manufacturing. Next slide. I'm sharing with you the, so to say, authority-approved quality control workflow for CAR T cell manufacturing, starting from incoming apheresis to transduction and harvesting. And the surface markers which are relevant to release the starting material and the 2 in-process controls are listed below. We call it panel A and panel B. Sorry. Good. Good. These quality control methods, which are shown here, are sufficient for the release of the product. And on the next slide, please. The authorities, we and others, are asking to do more characterization of the starting and materials and and the final drug product, meaning in this case the CAR T cells. We all want to generate more understanding which cells and which markers lead to long-term repopulation of CAR T cells, which cells are generating the clear clinical effect. So what we know today, we are going to the ideal cells are T cell memory cells or T cell memory stem cells. It seems, and maybe we make a quality attribute out of it in future, and it is very important to generate process workflows for CAR T cells, which in preference are generating these cells. If you are making T cell effector cells, you clearly see a beneficial effect in the patient, but this effect is not long lasting. So there are clear indications that T memory stem cells and T cell memory cells are these cells which do persist a long time in the patient and which are the cells which make the T effector cells to kill potential tumor cells in the patient. Next slide. Yeah, this slide shows you the challenge of quality control, how much time you need to do for one patient product. It's in the range of 20 to 25 hours. You need QC time. And then if you scale up, you can just see the difficulties in scale up. If you're running a small facility which makes 5 products a month,
Doctor,
you can manage it.
5 minutes.
Okay, good.
Time to finish.
Thank you. Okay, then the question is automatization. Next slide. And we were working on the automatization, and especially without going into detail, we worked on the automated gating of flow cytometric data. The analysis of flow cytometric data with the gating takes 10 to 15 minutes per analysis, and with an automatic gating strategy, you can reduce it to 1 minute, and therefore it's a big step ahead. Next slide. We developed a system based during the COVID time, supported by the German government to automate the quality control process. And this automation effort has resulted in the next slide, please. This slide is missing. You can go back. We developed an automated system to do flow cytometric analysis and also potency testing. And this system is now used, also being implemented with 2 industrial cooperation partners in order to streamline the whole quality control efforts. These are, so to say, the automation is implemented in sites which are able to manufacture 2,000 and more products per year. Okay, 5 minutes to go. I would like to draw you on regulatory answers to all these challenges. Next slide. In Europe, we have made the European Pharma Package, so-called. Next slide. And this is just to make sure these developments are under discussions. They are not, so to say, finally implemented. But nevertheless, I believe it is very interesting for the audience and the other regulatory bodies in this audience to see what is happening in Europe. Next slide. So the current framework is complicated because several legislations are responsible to manufacture, to regulate ATMPs. And the planned review is, so to say, unifying all these independent regulations into one medical product directive, into a master regulation package, which does include, so to say, is evening and streamlining all the regulatory frameworks for ATMPs. Next slide. And the objective is the objective of these regulations are shown in this slide, to have one single market for medicines in the EU, but budgets, affordability and availability and access to markets are clear targets of this objective. Next slide. And then there are also the regulatory bodies see that ATMPs cannot only be developed by big pharmas, but also by hospitals and by decentralized manufacturing efforts. And so the regulations is strengthening the hospital exemption, which is also discussed in several Asian countries. But it's also giving, as a first guidance, the possibility of decentralized manufacturing, meaning if a company, a hospital has one manufacturing permission, one product, it will become much easier to qualify additional sites to manufacture this product. And this will really enabling the large-scale manufacturing of these important drugs. Europe will also then implement a master file system, which is already established in the United States, and which is pretty much easing the regulatory processes, especially for raw materials and manufacturing tools. The environmental risk assessment is for genetic modified organisms, very important. In Asia, you might know it under the name of the Cartagena protocol, and this is also streamlined here. And like other regulatory bodies, regulatory sandboxes limited for a certain time are implemented. Yeah, and this is what I wanted to share with you. Again, I apologize for the technical issues, but you managed very nicely to show my PDF slides. I would like to, my great gratitude to you. Thank you, and now question and answers, please.
Yeah. Thank you, Dr. Ulf. Just allow me just to recap in bahasa. Jadi Bapak Ibu sekalian, Dr. Ulf ini memberikan kita suatu pandangan yang baru, utamanya mengenai point of care manufacturing. Apa itu point of care manufacturing? Itu jadinya suatu industri dari sel manufacturing tidak lagi base-nya di lab atau di industrial base, tapi di point of care, yaitu di rumah sakit. Dan yang Dr. Ulf tadi paparkan adalah gimana sih GMP-nya kalau itu ditarik dari industrial ke point of care. Dan terakhir ada memang around in EU bagaimana mengatur tentang hal ini. Jadi sekarang Bapak Ibu kita juga harus berpikir sebagai regulator ke depannya itu nanti seperti apa meregulasi produk-produk yang akan dibuat di rumah sakit untuk memfasilitasi ATMP ini memang tidak diproduksi banyak. Jadi satu batch, satu lot, satu pasien gitu. Yeah, so we are going to have to open the Q&A session. We would like to give like one or two questions first, and then Dr. Ulf can directly answer, so he can easily follow the your question. So this is a very rare change. So please, if you do want to know more about the GMP of ATMPs, the next future, then this is the time. Iya Bapak Ibu sekalian, tidak ada yang mau bertanya? Ya, gimana? So, Dr. Ulf, in the discussion of the new regulation of maybe you're aware about the new regulation that is being finalized about substance of original
Yeah.
or original human original, yeah. So is it like they also regulate these kind of things, like for example how into some what extent that the GMP compliance it's being brought into the like the hospital set of the facility. So I mean, is it already like in discussion that this is can be fully certified by the FDA, or this is remain on the hospital extension and not really oversight by the current FDA in EU? Can you just give your opinion on that? And
In the European Union, the hospital GMP is mainly following the established GMP for blood products and for stem cell products. So for blood products and for stem cell products, hospitals in Europe get, they have their legislation and they get GMP manufacturing permissions by the authorities, either locally or centrally, locally in Germany, Central East, for example, in France and Great Britain. And without such a GMP manufacturing permission, any product is not allowed to be manufactured. Then in the next step, having such a GMP manufacturing permission in place, the manufacturing of stem cells and other cells follows its legislation, which is established since many years. For CAR T cells, you need a separate manufacturing permission. Yeah, this is, but once it is established, it is not that, I can say it is not that difficult, but other people say it's a lot of difficulties. Yeah, so it is challenging if you have no background experience. It is straightforward if the facility and the people already know how to manufacture stem cells or other cell products.
So it's actually FDA is oversight this this small establishment in hospital. Is that correct? Okay, so they are subject to be GMP certified or certification, or there is a letter saying that this is a GMP certified? How is it work?
You need the GMP certification to, you need, the process is as follows. First, you do all the necessary work, mainly verification ones, to gain your GMP manufacturing certificate to manufacture. I stay now with CAR T cells, and then you can manufacture on a so-called one-hand principle for a limited number of patients CAR T cells, but then you have to move to IIT FDA approved, ethic committee approved protocols to to test your car product with patients. And the IIT protocol can then result into you transfer it to an industrial partner, you try to establish a point of care manufacturing permission. This is something the hospital needs to discuss with the local authorities. What happens after the initial IIT trial? There are several ways now open. In Thailand, for example, the hospitals, 2 hospitals are applying for a point of care manufacturing permission. In Europe, the Barcelona St. Paul University Hospital has in-hospital manufacturing permission by the European Medical Agency. So there are different models available now.
Okay, thank you. Is there any other question?
Major point,
Ada lagi pertanyaan yang lain? So Dr. Ulf, so following you that what you're saying that so every hospital establishment they should gone through this IND pathway. So the each product is actually registered as a they have a marketing authorization.
it is,
holder.
yeah, this is a good question. For your statement is correct for industrial pharmaceutical companies. For hospitals, a point of care manufacturing permission is not a commercialization of a product. It is, so to say, a personal prescription of a CAR T cell by the doctor for a patient. So what I do recommend is that industrial companies or hospitals who are interested talk with the local authorities, with the FDA, in order to stratify the entire development and what is necessary at which point. GMP, manufacturing permission, IIT trial, and what happens afterward. And there are several ways, especially in the emerging markets. You want to manufacture the product for an economical price, which can be afforded by many patients. Yeah. And therefore, it is very essential that hospitals work together with authorities to really define the way how to make access for these products to many patients.
Yeah. Thank you so much, Dr. Ulf. If there is no more question from the floor, or do you want to ask question? Somebody? No? From the GMP inspector's point of view? Nobody? Okay, then if it is not, thank you so much for opening our mind for what is really happening. Yeah. And we wish you a very pleasant. You also in the middle of the workshop, yeah. If I'm
Yeah, the workshop starts in 2 hours. Yeah,
okay.
yeah.
Okay.
11, it's now, the people are arriving now.
So
Thank you
wish
so much.
luck and enjoy the rest of your day, Dr. Ulf. Thank you for having sharing your experience with us. Thank you so much.
Thank you. Thanks to
Okay,
you.
thank you so much, Dr. Bethke, for your insightful presentation for today. Appreciation of your work, we are proud to present this certificate of appreciation. Let's give Dr. Bethke a round of
Thank you. Thank you very much. And I wish you a successful workshop.
Okay, thank you.
And hope to see you face to face soon. Bye-bye.
Okay, bye-bye. Thank you. Boleh kita berikan tepuk tangan sekali lagi Bapak Ibu?
Terima kasih kepada Ibu Santi yang telah memandu acara ini. Mari kita berikan tepuk tangan sekali lagi yang meriah untuk narasumber dan Ibu moderator kita pada hari ini.
Bapak Ibu yang terhormat, kita telah berada di penghujung acara workshop Elevate hari yang kedua. Mewakili segenap panitia yang bertugas, kami memohon maaf apabila ada kesalahan dan kekurangan. Terima kasih atas perhatian dan kerjasama peserta workshop pada hari kedua ini. Sebagai informasi, kami informasikan workshop hari ketiga akan dimulai pada pukul 8 waktu Indonesia Barat di tempat yang sama. Mengingat narasumber kita berasal dari USFDA, sehingga kami harapkan peserta dapat hadir pukul 7.50 untuk melakukan registrasi. Kami ulangi, workshop hari ketiga akan dimulai pada pukul 8 waktu Indonesia Barat di tempat yang sama, mengingat narasumber kita berasal dari USFDA, Amerika Serikat. Baik, sampai bertemu kembali di esok hari Bapak Ibu. Kami selaku MC mohon undur diri. Wassalamualaikum warahmatullahi wabarakatuh.
Seribu jalan di dunia, mengapa berpapasan bertemu dia?
Inginnya lari pergi tanpa kata, menyapa sudut mata, hafal rupanya. Lupa bahwa lupakannya mudah, tapi itu senyuman yang ku suka. Sepertinya sama, tatapan khas matanya masih yang lama. Kau ajak bicara, seketika kembali ku jatuh cinta. Sialan dia, sakitan air mata sia-sia.
Dari begitu banyak manusia, mengapa lagi-lagi dia yang ku cinta?
Malam hangat tapi ku terjaga, rindukan dirinya si pusat kecewa. Lupa bahwa lupakannya tak mudah, tapi menyangkut dia ku tak bisa. Sepertinya sama, tatapan khas matanya masih
yang seragam biru,
kau ajak
biar pulang sih.
bicara, seketika kembali ku jatuh cinta. Sialan dia, sakitan air mata sia-sia. Walau saat tepat di titik kelemahanku,
Letakkan di hatiku
terbaik tetap selalu ada singgasana, tempat untuknya. Sepertinya sama,
khas matanya masih yang lama.
ajak bicara, seketika kembali ku jatuh cinta. Sialan dia, sakitan air mata
Untuk teman-teman PB ditunggu di depan panggung, sudah dipanggil oleh Ibu Katim untuk foto.
Ibat mana Ibat? Diar ikut Diar. Iki. Udah Diar udah. Ibat. Oh iya Musafak. Ayo Musafak.
Yes.
Ham. Ayo ditunggu dong teman-teman. Ayo. Rumah gue jauh nih. Ayo Mas Mus cepetan. Siapa lagi yang belum? Iki, ayo merapat. Mbak Diar udah ya? Mbak Diar udah. Mas Ibat. Mas Ibat mana ya? Oh itu dia. Mas Mus, ayo jangan pura-pura
Badan
sibuk.
kolagen bikin hingga 10 kali lebih kuat lawan rusak. Kondi Koconai. Suplemen conditioner.
Akrab dong, rangkul dong, rangkul.
Cek, cek, cek. Belum, belum. Belum, belum. Baik. Hmm. Ada kayaknya.
Apa yang dia buat gitu? Apa sama yang